Okay, let us start. Merci. Please unmute yourself and you will start a call up one by one.
Yes.
Unmute yourself. Merci. Merci, unmute.
Yes, ma'am. Yes, ma'am. I unmuted myself. Good afternoon, everyone. After a technical session, now we have entered into the presentation session. I welcome you all. I firstly would like to thank all the jury members for taking your time to judge the sessions, and instructions to all the delegates who are presenting. 5 minutes will be given for your presentation, and 2 to 3 minutes will be for the discussion. If there is any question posed to you from the judges, then you need to answer during this discussion session. I hope this is clear to all the delegates who are presenting. Let us start with the first person, that is Debashri Borthakur. The first participant. If you are ready with the presentation, can you please share it?
Unmute your video first. Debashri, I have given the co-host for you.
Debashri Borthakur.
Am I audible?
Yes, you are.
Yes.
Actually, I was trying to unmute myself, but it needed some permission. So I'm late. I'm extremely sorry for this. Can I share my screen?
Yes, you can share.
Yeah, thank you so much. May I know if my screen is visible to everyone?
Yes, ma'am.
So can I start presenting? Can I start presenting?
Actually, go for full screen.
Yeah,
Go
yeah.
for full screen. Okay.
Okay, sure. Is it visible now?
Yes, it is. Go ahead.
Okay, fine. Good. A very warm good afternoon to all the delegates present here. So my name is Debashri Borthakur. I am a research scholar from Tripura University. So my topic of presentation is health benefits of fermented foods of microbial origin from Northeast India. Coming to introduction, as we all know, fermented foods have inhabited an essential role in socio-culture and many non-identical socio-culture and many tradition of non-identical tribes. Fermented foods are beneficial as they are cost-friendly and they are also good for health. Drying and salting are two basically important methods we are using for producing a fermented food. Fermentation in fermented food, normally fermentation is carried out by microbes that basically changes the raw materials into simple. This individual fermented food is associated with unique individual microbes which give them a proper flavor as well as also enhances the nutrients like proteins, amino acids, fatty acids, and other nutritional constituents. As my topic is specific for Northeast, so basically Northeast consists of 8 states, namely Assam, Arunachal Pradesh, Manipur, Meghalaya, Mizoram, Nagaland, Sikkim, and Tripura. So the fermented food generally contains lactic acid bacteria, and this lactic acid bacteria are live bacteria. So we also call them probiotic. So this is one species of lactic acid bacteria are of different types like Lactobacillus leuconostoc, Lactobacillus fermentum, Lactobacillus casei, etc. So these probiotics are basically the live microorganisms which are inoculated into the food to improve the intestinal balance of the food as well as to improve the flavor of the food. Several studies have reported that insertion of probiotics to food boost up the health benefits such as cure to lactose intolerance. This is a disease where a person is not able to digest the lactose, that is milk sugar present in a dairy products. But it also helps in better gastrointestinal functions. It also improved immune system, reduction of serum cholesterol, and also lowers the chances of colon cancer. Coming to diversity of ethnic fermented foods in India. So apart from Northeast, the fermented foods are largely used in other parts, other states, like some of the fermented foods from other states are babru, which is a fermented food of wheat or rice flavor, and it is from Himachal. Hakua, that is from Darjeeling Hills. Appam, this is a fermented rice from Kerala. Selroti, this is a rice wheat flour from Northeast India, as well as it is also available in Uttarakhand, Himachal Pradesh. Agyas, it is a fermented soybean from Arunachal Pradesh. This is Bari, it is also a fermented soybean from Sikkim. Bekam, this is also a fermented soybean from Mizoram. Dhokla, this is a Bengal gram and rice, fermented Bengal gram and rice mixture, and it is from Gujarat. Hakumata, this is a fermented soybeans from Manipur. So the kinema is also a soybean from Sikkim, Darjeeling. Peron, this is also a soybean from Arunachal Pradesh. Tangrimbai, this is also a soybean, fermented soybean from Meghalaya. Varai, this is a black gram, fermented black gram from South India, almost in all states of the South. Yani Perung, this is a fermented soybean from Arunachal Pradesh. Chura, this is a fermented milk from Ladakh. Dahi, we all know it is available in all parts of the country. Lassi, this is also available in all parts of the country, and it's a fermented milk. Filu, this is yak milk. It is available particularly in Sikkim. Khalari, this is a goat milk available in Kashmiri. Paneer, this is also available in all states. Coming particularly to Northeast, Imao, this is a fermented alcoholic beverages from Assam. Humao, this is a fermented rice. It is a drink kind of thing. This is from Assam. Ipo, it is a fermented rice from Arunachal Pradesh. Kim, it is a fermented wheat from Himachal Pradesh. Marcha, this is from Darjeeling Hills. Vekur Pitha, this is a rice leaves of some local plants, and it is specifically from Assam. Apong, this is a fermented rice from Assam. Atingba, this is a fermented rice from Manipur. Buja, this is a fermented barley from Ladakh, and this is chuli, a fermented apricot from Himachal Pradesh. So these are also some fermented foods specifically from different parts of the country. And coming to fermented fishes, this is Godak. This is a Puntia species, and it is from Tripura. This is a fermented fish. Chucha, this is a small fish species, and it is from Assam. Nuchi, this is a fermented fish from Darjeeling and Sikkim. Hentak, this is a fermented fish from Manipur. Karati, this is also a fermented fish from Assam and Meghalaya. Nakamba, this is a fermented fish from Manipur. Nagrain, this is a small fermented fish from Assam. Small fishes, these are Sepa, this is also fermented small fish from Tripura and Nagaland. Shidal, this is a fermented puntis from Tripura and Assam. Tungta, this is also fermented fish from Meghalaya. Sidra, this is also fermented fish from Eastern Nepal, Darjeeling, Sikkim, and Bhutan. Coming to some fermented meats, Jangkap, this is a fermented buffalo skin from Nagaland. Lang Karyong, this is a fermented beef from Darjeeling. Lang Sachu, this is a fermented beef from Darjeeling. Lang Chilu, this is also a fermented beef from Sikkim and Darjeeling. Fak Karyong, this is a fermented pork from Darjeeling and Sikkim. Bangjinam, this is a fermented pork from Nagaland. Sukaku Masu, this is a fermented meat from Darjeeling. Yak Chilu is also a fermented meat from Darjeeling, that is yak meat. Yak Sachu is also yak meat from Sikkim. Apart from this fermented dishes, these studies have reported that microorganisms are also able to introduce certain flavor to the fermented food. So these are some flavorous compounds which are produced by microorganism by producing an active component, like almond type flavor. Almond type flavor is produced by benzaldehyde, which is basically reported to produce by Echinoderma banjonium. This is a fungus. Apple and pineapple flavor, this is also evolved due to the production of butyric acid by Clostridium butyricum species.
Please conclude. Dibashri, please conclude.
Okay, ma'am, sure. I'll do that. And these are some important enzymes which also helps in flavor production. Coming to the health benefits of probiotics, currently many studies have reported that beneficial effects of probiotics on human health as well as on the other human health. So coming to improvement in human health. It basically boosts up the immune system. It improves digestion. As I have already told you, it helps in tolerating lactose intolerance. It balances the good bacteria in the intestine. It also supports the liver health, lowers cholesterol, keeps heart healthy, better breath, makes skin healthier, decrease tumor activity, helps treat diarrhea, supports mental health, and it also reduces the tooth decay. Apart from this, there are we cannot include all organisms as probiotic. There are certain characteristics of probiotics, like probiotic. Sorry, is my slide visible now? My slide's visible now?
Yes, visible, but your time is up. That's
Like
why I stopped it.
Probiotic must survive along the passes of probiotic must survive along the passes. Okay. Can I stop? Shall I stop now? Probiotic must pass through the gastrointestinal tract.
I think we can go for the questions now.
Yes.
See, your time got over.
Okay.
Yeah.
Thank you so much.
Actually,
So I
you
would like
put
to
in
welcome
lots of time in introduction, like rather than coming to your thing. And it is like a review kind of thing, isn't it?
Yes, ma'am. It is
You're
like
doing your
we have
PhD. Are you doing your PhD now?
Ma'am, actually, because of this, because I have just started my PhD, we are done with the coursework, and also we have done the isolation part. From this fermented foods, we have isolated microorganism. So, like, that is why I have given the presentation on health benefits. Not much work is done. So only till health benefits we have, yeah.
Okay, I feel that especially for this kind of conferences, you could have presented something related to research work rather than going for a review. It's fine.
Actually, ma'am, I was about to, but as no much work is done, so I didn't present.
Okay, others can ask questions.
Yeah. Secondly, Debashree, you have your first slide, second slide, and last slide. You see the references how you have quoted. The references should not be quoted in that way. No, since you are a research scholar, you should have quoted the references correctly. Okay, that is first thing. Then you have said the health benefit is lowering cholesterol. How does fermented meat lower cholesterol?
Sir, not fermented meat, because we, apart from fermented meat, we have fermented greens also, and these greens are exclusively noted for reducing cholesterol. As the microorganisms which are involved in fermentation, they produce certain enzymes. So this actually helps in removing the bad cholesterol from the body.
Okay. Okay. So yeah, over to others.
Fermented meat is also, sir, very specific. That is not used in other states of Assam. It is specific to Sikkim only. Fermented meat.
Yeah, but you said there are health benefits of this this food. That's why I'm asking.
Yeah, I actually need to mention that. Yeah, that is for fermented.
And are there probiotics in that food?
Green.
Are there probiotics in that fermented meat? Are there probiotics in that?
Sir, in
yeah, you should have removed that.
Are there, but
yeah, which bacteria are there in that fermented meat?
Like in meat you are asking, or
yes.
Like in all fermented food?
No,
so like
in meat.
We have, I'm telling you, so we have isolated some bacteria from fermented fish. We have not done meat part. So from fermented fish, what we have seen that mostly they are gram positive bacteria, because we have done till gram positive only, gram staining only. So we have seen that maximum of them are gram positive bacteria. Yet we need to do biochemical test, and we need to identify the strain also by 16S rRNA identification technology.
Okay, fine. Okay, over to others.
A small suggestion from my side. The title of your poster and the paper is not.
Ma'am, your voice is breaking. Ma'am, your voice is breaking. Can you be a bit loud, please?
Is it clear now?
Yeah, it's fine now.
Yeah, the title of your talk itself should have given a review article, because that is giving a wrong impression that you have done your bench work. That is one small suggestion I would like to give. And you have said their fermented methods like drying and salting. I don't think they'll come under the fermented methods, which you have put in your sentence like that, if I'm not wrong.
Ma'am, can you please repeat once, drying and salting part?
No, you have given them as the fermentation methods. If I am
Yes.
drying and salting are fermentation methods.
Ma'am, actually, traditionally they are using drying and salting only in this some specific states of Northeast. They are not using very advanced technology since they are from remote areas. So they use only drying and then they put it in a salt solution, keep
But
it
in
for a
the
long
salt,
duration.
no, salt will prevent the microorganisms growth, no?
Yeah, ma'am.
It comes as
Actually, it helps.
fermentation. No, I'm
Yeah,
just.
ma'am. It actually it promotes plasmolysis, but they used it as a preservatives, not
Yeah, preservative,
preservatives of.
not. Yeah, they are preservatives, not methods of fermentation. That's what
Yeah.
I understand. Yeah, maybe you can just look back at the sentence. And whenever you are presenting this kind of work, you need to give in the title itself as the review article. Okay?
Oh, yeah. I'm
that is
extremely
always.
sorry, ma'am.
No, it's not required. Sorry and all, just to make you understand because you are just in the beginning of your research. Yeah.
Thank you, Debashri. All the very best. Next participant, N. Raghavendra Babu. Please carry on.
Yeah. Is my screen visible, madam?
Yes, it is.
Yeah. Okay. So good afternoon to the delegates and the jury members and organizers of this conference. Myself and Raghavendra Babu. The title of the presentation is Synthesis, Characterization and Antitubercular Evaluation of Tetrahydrotetrazooloquinazolone Derivatives. So coming to introduction, generally tuberculosis, it is a COVID, it is a contagious disease that is major source of illness, and it's leading to cause loss of mortality in the world. So this is going to cause of death from single infectious agent until the coronavirus pandemic, ranking ahead of HIV as well as AIDS. So this main organism is Mycobacterium tuberculosis that is transferred through air when the patients exhale, when the patients with the disease, when they are going to exhale the bacteria, that is by coughing. So here, generally it affects the lungs, that is a pulmonary tuberculosis, and it can also affect the other parts of the body. So generally drug-resistant Mycobacterium tuberculosis strains, they particularly has confounded treatment procedures and raised fears that tuberculosis will once again become an incurable illness. So it's vital to find new drugs that work in a different way from the ones which are already in the use. So generally, there is resistance also for, but the resistance has been developed for those drugs. So when you go for tetrazole cycle moiety, it has various diversifiable activities like antitubercular, bacterial, antifungal, antiviral, anti-inflammatory, anti-ulcer, anti-hypertension are there for tetrazole activities. And whenever you go for quinazoline containing compounds, they are usually nitrogen containing compounds, and they are also having various anti-tubercular, antifungal, anti-cancer, and anti-HIV activities, anti-HIV, anti-inflammatory, analgesic, antibacterial activities. So when we have gone through the literature review, so the majority of them have synthesized some compounds like 6,6-dimethyl tetrahydrotetrazooloquinazolone derivatives. So we try to synthesize new derivatives. That means we try to fuse this both tetrazole and quinazole. So when we try to fuse both tetrazole and quinazole, so we have come with new compounds that were very much active in tuberculosis in order to kill the tuberculosis bacteria. So whenever we come with the methodology, the synthesis of compounds, so here when 5-aminotetrazole, when it reacts with the dimidone, so this leads to, in the presence of isopropyl alcohol, it leads to formation of compound 1. When this compound 1 is
Sorry, but the screen is not moving, sir. You are thinking it
not
is moving?
madam.
Yes.
Not moving, madam.
Yes, it is not
You
moving.
have not made slideshow. Animation not activated.
Yes, sir, I made it. Sir.
It is in the first slide only, sir.
One minute, madam. 1 minute, please.
Reshare it.
Yeah. Please give me the time, sir, please. Now it's okay, sir.
Yes.
Methodology. Hello.
Yeah, it's visible now.
Yeah, you can go ahead.
Yeah. The 5-amino tetrazole and bendimidone are both of them have treated in the presence of isopropyl alcohol and 10% iodine has been used. It leads to formation of compound 1. So the formed compound 1, when it is reacted with different benzaldehydes in the presence of isopropyl alcohol as a solvent again, that leads to formation of different tetrahydrotetrazole and quinazolone derivatives have been formed. So majorly when I have reacted this bromoacetophenone, so this has been fused with NH. So the H has been replaced with this particular ring that showed very promising compounds with good antibacterial activity. And this spectral data, the formed compounds were confirmed through spectral data through NMR, IR, and mass compounds, mass spectral data. And whenever I done molecular docking, so by taking a protein like thioestereose domain complex inhibitor, that is TAM5 with PBD ID of 5V41. So when we have compared with that of that particular protein, so seriously, our compounds were having more docking score, like F1, it has minus 12.32. F2, it has minus 10.16, F3, minus 11.98. So like this, we have most of them, 8 compounds are having more than that of this ligand, that is protein ligand. So this particular table has been summarizes that particular docking score. And in this particular figure, this F1, they gave a docking score of minus 12.32. When this has been docked with the Autodock software, we have found these two F1 as well as F4 compound docking scores compared to that of that particular protein. And coming to, I think, major part, that is antitubercular activity of these compounds. So whatever this compounds antitubercular activity, this particular activity was tested using microplate Aramer blue assay. And this is non-toxic method. It is a thermally stable reagent and has a high level of correlation, that is, which is proportional to the bacteric radiometric methods. So here 96 well plate was taken, and this particular compounds were tested with the Aramer blue reagent, that is 10% twin 80 was added, and which are incubated for 24 hours. And the blue color in the well indicated that the bacterial growth, whereas pink color indicated the growth. That is, the blue color is indicating there is no bacterial growth, that is inhibition of the bacterial growth. So the minimum inhibitory concentration is defined as the nothing but the lowest concentration of the drug that prevented the color change from blue to pink. So when you compare the results, these are the results of the antitubercular activity. So compound F4 and F7. So F4 and F7, they are having Mycobacterium tuberculosis, they are inhibiting the strains up to 6.25 microgram per ml, where F2 and F5 are inhibiting at 12.5 microgram per ml. So
please conclude, sir. Please conclude.
Yes, madam. Okay. And teratogenicity. Conclusion. So first we have synthesized some compounds by conducting the docking studies, and later at different concentrations like 6.25 microgram per ml, and F4 and F7 compounds were active against Mycobacterium tuberculosis strains, and F2, F5 were effective at 12.5 microgram per ml respectively. So these compounds were subjected to the toxic study also. So when I have conducted toxic studies in zebrafish model, F10, except F10 compound, all were free of toxic nature. So getting activity is not at all important, but whether the particular drugs are safe or not, that also we need to see. So when I have conducted with zebrafish model, all compounds were safe except F10 compounds. So I'm very much, these are the references, and I'm very much thankful to the Department of Microbiology, Palamur University, for giving me this opportunity to present my research work. Thank you.
Yeah. Hello.
Yes, sir.
Yeah. Am I audible? Yeah. Okay.
Yes.
In your docking scores, you have shown that F1 is having highest docking score.
Yes, sir.
Yeah. Then when it comes to the activity, actual activity, that is the practical work in silico and in vitro. In vitro studies, there is a difference. Can you explain why there is a difference between both of them?
So, sir, whenever this is through docking, this is only a computational technique, sir. Generally, when we go with docking, so we are getting one score, but exactly that particular score is not going to be reflected in the activity sense whenever we go for with the microplate Aramer blue assay technique. So that is the reason. So exactly docking, whatever the result we are getting, that we cannot expect in the original activity whenever we are going to do that.
So it's not required, or is it required?
Yeah, docking is required, but to know what extent the compounds are showing. So it may be showing minus 12.1 in the docking studies, but when you come to F4, it has been matched. Not all it depends upon the moiety, whatever we are using, like fluoro group, like chloro group, like different halogens, electron donating groups, electron withdrawing groups. It completely depends upon the particular group we are going to keep insert in that particular structure, sir.
Okay, fine. Over to others.
Okay, sir.
So what is the mode of action of your compound, which you have synthesized? What is the mode of action, yeah, against this Mycobacterium tuberculosis? Like how it acts?
Yeah, no, madam, it is a technique. Whatever I have used is H37RV strain technique I have used, madam. So first they develop the growth of the microorganism. Then they they make my compound into different dilutions, and we have added at different levels. So at particular microgram per ml, that particular growth has been inhibited.
Yeah, but it might be acting in a particular way, isn't it? There's something wrong. Hello? I think it got disconnected for him.
I am there, madam.
Okay, but yeah. No, but you need to understand like how it acts, isn't it, against Mycobacterium?
Yeah.
Yeah, that is important. And what was your positive control? Did you compare it with the positive control?
Yes, isoniazid, rifampicin, and isoniazid and rifampicin. With both I have compared, and so they were active at 1.8 microgram per ml, but my compounds are active at 6.25 microgram per ml.
Yeah, so they are.
Yeah, it's very near to the standard compound. But in future, what we can do is, if you incorporate still other electron donating groups, electron withdrawing groups into the key structure, so we can slowly achieve the target compounds. Okay. That's what I need to say. That is a future perspective of the work.
Fine.
So, sir, am I audible?
Yes. Yeah, madam. Yes, madam.
Yeah, sir, did you use the same concentrations for the model organism toxicity and the antimicrobial activity? Did you maintain the same concentrations?
Yes, madam. Yes, madam.
Yeah. So
that was done on zebrafish, madam.
Yeah, that's what. So yes, the same level what you have tested in zebrafish would be able to show the antibacterial activity.
Yes, madam.
So did you follow any green synthesis methods to synthesize
no, actually,
the compounds?
No, madam. Green synthesis, actually, the third step, whatever I have used now, that comes under green synthesis only, because just I have triturated in the mortar and pestle by taking two compounds. Just I have triturated in the mortar and pestle, the compound has formed, which was confirmed with spectral data. So I didn't go with the microwave technique, or I didn't go with the solvents in the last step. But two steps I have used solvents itself, madam.
Okay.
Sandipta madam, please welcome to this session. A warm welcome to you, ma'am. Please join in the panel. Sandipta ma'am.
Thank you. Yeah. I was at a wedding, so by the time I joined, it was after 2 o'clock.
Okay.
But I've been trying to talk, but I could not unmute myself. Anyway, please continue. I will ask questions in the next one. Okay.
Okay. You join next too. Thank you, ma'am.
Muskan, you can start your work.
Thank you, sir. Thank you, madam, for giving the opportunity. Thank you so much.
Thank you. All the very best. Next participant, Muskan Jain.
Ma'am, is my screen visible?
Yes, it is. Continue.
Okay. Ma'am, I'm putting it in the slide. Yes, ma'am. So good afternoon, everyone. My name is Muskan Jain, and I'm pursuing BTech from Biotechnology Department from Amity Institute of Biotechnology, Uttar Pradesh, Lucknow. So the topic of my presentation is metagenomics study of breast cancer. So here in this PPT, I am going to cover that what is metagenomics, what is breast cancer, and what are the materials and the methodology I have obtained for, I have followed for obtaining the results, and then the results. So first of all, what is metagenomics? Metagenomics comes under the field of bioinformatics, and it is the inspection of hereditary material recuperated straightforwardly from environmental instance. And the broad field may also alluded to as natural genomics, ecogenomics, or community genomics. Metagenomics gives a culture-independent perception about microbial communities, and it has come out as a powerful core since most microorganisms can't grow in a pure culture, and that culturing can't capture the full spectrum of microbial variegation. The term metagenomics was first employed by Joe Henderson, John Clardy, Robert M. Goodman, Sean F. Brady, and others, and was displayed in the year of 1998. Now, what is breast cancer? So, breast cancer is a type of malignant cancer. That is, it can spread from one part of the body to the other, and it is the swelling structured in the breast cells. And after the skin cancer, breast cancer is the most broadly accepted malignancy that was analyzed in the females. So, here are some signs and symptoms which show that if a person is having breast cancer or not. So the first symptom is lump on the breast. The second is skin dimpling. The third is change in skin color or texture, change in how the nipple looks like pulling it off the nipple, and the last is clear or bloody fluid that leaks out of the nipple. For this research was I have studied or worked on two databases. That first was ENA database. So ENA database is a European nucleotide archive, and it provides a comprehensive record of the world's nucleotide sequencing information, covering raw sequencing data, sequence assembly information, and functional annotation. And we can search in this database by searching the terms or with the accession number. And this image is showing the homepage of ENA database. Now, I have also worked on the Galaxy database. It is the second database which I worked on, and it is an open source web-based platform. That means you no need to download it from the Google. You can directly work on it by signing it in it. So yes. So I have used certain tools that were the part of the Galaxy software only. So the first tool which I have used was quality control or FastQC. So FastQC aims to provide a simple way to do some quality control checks that if there is any problem in your data, then it will show that you have problem in your data. You need to re-download it or re-upload it. And the second tool which I have used was Trimomatic. So Trimomatic is a tool that was used for trimming of particular data. And the result of Trimomatic was the input of Kraken 2. Kraken 2 is a tool that was used to assign the taxonomical labels to your data, that to which class it belongs to, or which kingdom it belongs to, or which phylum. Now, the result of Kraken 2 was the input for Convert Kraken. Now, Convert Kraken classifies your data, and it assigns the NCBI taxonomy in it. And lastly, I have used the Corona pie chart. Corona pie chart was the representation of the organism which were there involved in the breast cancer. So first of all, I will present you the result of FastQC. So FastQC results were depicting in the form of graphs. The first was per base sequence quality. The second graph was showing per sequence quality scores. And the third one was depicting the GC content in your data or in the sample. The second result was Kraken 2 results. So the first column is showing that that is representing U or C. It is showing that your data is classified or it is unclassified. And the
Kindly
second,
conclude.
yes, sir, only 2 to 3 slides are left. So this was about the Kraken 2 result. And this is the convert Kraken result. So here in the table, you can see that the data is classified into subkingdom, kingdom, and phylum, class, etc. And this was the final result, that is the corona pie chart. You can see that the organisms are divided in the form of chart, that in how much percentage they are present in your sample, that is in the breast cancer. So you can see that the 2% of nematosora is present, or pyrognematia is present in the amount of 4%. So this is the final result in the form of corona, and in how much percentage your organism is present in a sample. So this complete project is to analyze the taxonomical function analysis of the metagenome of breast cancer, and a number of varieties were found in the analysis. So these 5 bacteria were basically, these 5 organisms were basically found in the high percentage, that is actinobacteria, bifidobacterium cuniculi, murine leukemia virus, human endogenous retrovirus, and pezizomycotinum. And this was it about my research work. Thank you, everyone.
Muskan, what is the source of your data? From where you collected the data?
Ma'am, I have collected the data from the database, that is ENA. From ENA database, I have downloaded the sample from there.
Okay, so it is only taking the samples from a particular database. It's
Yes,
not you have collected it from some patients or something like
no,
that.
Ma'am. No, ma'am. Actually, metagenomics is a branch of bioinformatics. So basically, we have to research on computers only, that what is the percentage of certain microorganism present in that sample.
So there's no need of sample collection. It is already existing data which you are using.
Yes, ma'am. It was already existing.
People go for, see, metagenomics. It doesn't mean that you are taking only the data from which is existing, isn't it? Like you can also collect the data. The samples can be collected and it can be utilized. Like you can go for like comparing the databases and all, isn't it? But anyway, because you are a BTech student, so for you, I think at this stage it will be, it's fine. Okay.
Yes, ma'am. Ma'am, it was the comparison of the infected person and uninfected person. The person who was having the breast cancer and the person who wasn't having the breast cancer.
Control. So how many, basically the sample size or something, have you taken like that?
No, ma'am. No, ma'am. It was already there in the file which I have downloaded from the ENA database. Ma'am, the main task of my project was to see that how much of percentage was present in the patient who is having breast cancer, and the normal microbiome of the breast of the lady or person.
Okay.
Yes.
Muskan.
So, Muskan.
Yeah, hello.
Yes, sir, you're audible, sir. Yes,
yeah,
sir.
Muskan. Yeah, the work the title itself should have been like, it is a computational analysis of the metagenomic this thing. Like it looks like, I mean, you have done some metagenomic work on the samples, and then you have put it there. Like now in a database, whatever data you got, whatever data you have retrieved, okay, that is the data which someone else has done it already. Okay, and
Yes,
then
sir.
they have
Yes.
deposited in the database.
Yes,
Okay,
sir.
so
Yes.
the metagenomic analysis has been done by someone else, and they have deposited the data there. Based on the deposited data, you have done a computational analysis. Okay, so the title should have been the computational analysis. And as madam already said, metagenomics is not bioinformatics, or it's not only deals with bioinformatics. It has so many other components. You need to do the culture and all the other things. Okay, you need to isolate the sequences, then give it for sequencing and all. Anyway, okay, thank you, all the best.
Thank you, sir. Thank you, sir.
So, Muskan, I have one question.
Yes, ma'am.
So the samples which you are saying, right, the metagenomics which you are talking about.
Yes,
So
ma'am.
where did the sample come from the patients? Which part of, is it like part of the lump, breast lump? Is it the skin, or is it some secretions from the breast? I did not understand.
Ma'am, sample is generally collected from the fluid only. Ma'am, then only we can study its microbiome or what are the microorganisms involved in it.
Yeah, but most of them were viruses, right? What they got were viruses. So how is it about bacterial diversity? Because we could not see much bacterial strains in that.
Ma'am, it was showing whole organism that was present or involved in causing the cancer. Bacteria also, viruses also.
So what is the conclusion? Okay, we got them. See, there are millions of breast cancer patients. Let's say these are the ones. So what is your conclusion that, okay, if these are present, maybe many cancers are anyway caused by viruses. So what is the conclusion from this study? How do you think it will be helpful to study breast cancer?
Ma'am, first of all, I came to know about that what are the signs and symptoms of breast cancer and what species or what organism are responsible for the breast cancer and the microbiome was.
Okay. So you are a BTech student, as madam has also said, but I don't know if actinobacteria, actinomycetes or whatever are responsible for cancer. We cannot know from this data, right?
Yes, ma'am.
Yeah, even bacterium and all, really, that is the diversity which is present there. That's all, I think.
Yes,
yeah, anyway, she is too young to, like, I think it is okay. We just attempt.
Exactly. Yeah.
Yeah.
It will be good to infer something from
Yeah,
the diversity.
true. That's true.
And also, I feel sample size one need to know, because based on that also you will be able to derive the data and apply that. Because in 100 samples, the data would be different. In 1,000 sample, the data would be different. Maybe you can focus on that.
Yes, ma'am. Thank you, ma'am. Thank you, everyone.
Thank you, Muskan, and all the very best. Next participant, Dr. Jeevan Chandra. Sir, are you ready with the presentation? Please carry on.
Thank you, madam. My presentation is visible to all?
Yes, sir, it's visible.
Make it full size.
Go to slideshow, sir.
Good morning, my dear viewers and organizing committee of Palambur University. They are presenting today conference on microbial pathogenesis and recent advances in diagnosis of Omicron. I thought of this topic is somewhat informative for the upcoming generation and students. So I have selected this topic.
Jeevan Chandra sir, please unmute yourself. Increase the volume, sir.
Okay, madam. I have
increase the volume, sir. You are not audible.
For us, it is audible. For us, it is okay.
Okay, madam. It is okay.
This topic will give you as an information regarding how the virus is evading from the immune response and become more virulent.
You can carry.
So the emerging viral disease always thought to be a problematic one for the public health purpose. So in microbiology view, it is very important to explore the molecular dynamics of the evolutionary track of SARS virus that will give us an information to address some peculiar mutations which are prevailing in the spike site that could be addressed in bioinformatic way. So that's why I have selected this topic for a presentation. So since last 3 years, we have been struggling a lot with the severe acute respiratory syndrome, SARS virus. So it has been alarming the incidence and the spread of the virus among the population. So what make the virus to be very virulent? Such a way, I have selected this topic. So far, more than what to call every day, there has been 2,000 around 1,000 incidents of cases are there in Telangana. All over the world, the pandemic situation has been arised. So the virus changed its morphological features. That's why it is evading very quickly from the past. So on November 24th, in South Africa and Botswana, a new variant was erased. That was beta B1.1.529 is nothing but a Omicron variety. The WHO also considered this variety as a variant of concern. So one peculiar feature is noted here. So later, now in Telangana particularly, the migrations and air traveling also highly affected the incidence of Omicron. Such a way, I have selected this topic to explain what are the peculiar features and molecular basis of the evasion mechanisms. So what do you know about the Omicron variety so far? The Omicron variety is nothing but a previously reported coronaviruses. It was first emerged in South Africa, then to Botswana. Later, it was traveled to Israel and Belgium, UK, Germany, Italy, China, and Australia, Netherlands, then Czech Republic, and then US. Now we have been reporting this virus in India itself, in Telangana also. So it is associated with several risk-associated factors because of it has a high replication, higher infection rate, and other aspects. So coming to the Delta variant and Beta variant, the Omicron has become more surged infection incidences on November 24th till now. So what made this virus to be evaded from the immune mechanisms? Such a way, we have to cite into the structural organization of the virus. The virus spike protein plays a major role where the receptor binding site and RBC receptor binding site and other related informations will give you information regarding what type of peculiar mutations that are going to be occurred in the viral site. So this spike is about 1273 nucleotide length, but RBD domain and SD2 SS domains are very important for consideration of the binding activity. So let us discuss about some immune mechanisms the Omicron has adopted. So as we know that it is a heavily mutated virus, and it acquired more than 22 potential mutations on the spike, which have changed the conformation and affinity towards the ACE2 receptor. So due to increased affinity of ACE2 receptor, which leads to transmissibility as well as the mutations prevailing on the spike protein. So the spike protein from the alpha variety, first it is showing that the amino acid aspartate at the 614 amino acid site was changed to glycine. Later, it was emerged into very effective pathogen. So coming to mutations on Omicron, more than 32 out of 50 mutations are just present on spike protein itself. So they are the notable factors which are able to carry out several affinity towards the ACE2 receptor. These all are the some of the amino acid changes, and very important. Red color one is very important. They are going to affecting the ACE2 receptors. Let us discuss about a last and very important topic: how malt mucosal associated first line defense has been first blocked. This what happens when the double stranded RNAs and single stranded RNAs are invading into your host cell. Now the RIG proteins and MIDAS proteins are going to be activated. RIG protein is nothing but a retinoic acid inducible gene product, is nothing but a pathogen associated molecular pattern recognition receptors, which are going to be upregulated, and toll-like receptors have been involved in the process, and they underwent into nucleotide synthesis. That means nuclear trafficking was also getting activated. Interferon gamma, interferon alpha, interferon beta also going to be produced. The mechanisms was most important. They are able to distinguish between the host RNA and viral RNAs. But what happens? The MIDAS RIG1 protein, especially very important to recognize the host as well as the self invading viral genomes. So what happens? The structural features which are very important to recognize the invading viruses. So what happens? Most of the viral proteins are going to be circularized. That's why the ATPase activated helicase systems are not promoting the system. Finally, the Midas protein was unable to recognize this invading RNA. So finally, what happens in the alveolar macrophages? They are going to be accumulated more and more. That leads to cytochrome storm, which is foremost important one. That's why our natural killer cells and toll-like receptors as well as the interleukin 6, they are going to be improved along in enormous quantity. Such a way, these all are lead to cytokine secretion profiling, and the infected cell has to be enhanced the recruitment of myeloid immune cells, natural killer cells, which in turn produce more cytokines, creating an inflammation situation which is going to be damage the lungs. So what happens? The signal cascade systems are targeted by the coronavirus proteins, and innate antiviral sensors can regulate the coronavirus invasion and sensing the cytosolic endosomal viral RNAs. Activation of virus sensors and triggers the viral evasion mechanisms. So these pathogen recognition receptors are also not working well. They are also blocking the natural killer cell recognition, T cell recognition, and other important features. Such a way, these all are leads to evasion of the immune mechanism. So another more important feature is that rapid viral RNA replication in innate immune antagonism. That is also recognized by this virus. So rapid viral replication and non-structural protein leveling contribution by sequestrating the double-stranded DNAs, and more important one, epitope masking and the induction of less endogenous double-stranded RNAs, which are all contributing to pathogen-associated molecular pattern to improve the innate immune stability, and augmentation of early protein expression in main innate immune antagonism contribute more to immune evasion, and helicase activity also going to contributing for replication. So the transmission, finally the Omicron variety, the transmissibility has increased more and more, which leads to dangerous pandemic situation. So these are some of the mechanisms. Even the convalescent sera also not working well on this one, but it will protect you about 50 to 30%. So finally, my conclusion. So evasion by interferon evasion mechanism and preventing viral immune evasion by contributing to enhancing viral clearance, but lessening the immunopathology. While major diverse force for this one, cytokine storm in COVID-19. So interleukin 6 has both pro-inflammatory and anti-inflammatory properties. So this will give you the most important feature for pathology. So inhibition of IL-6 is very important to elucidate the mechanism. And at the same time, research should be focused to both convalescent sera and early strains of Delta infected patients could have demonstrated very low capacity of neutralization. So that's why the mutations are very dynamic and they are contributing for evasion as well as the transmissibility of this virus. So thank you very much for providing this opportunity.
Dr. Jeevan, you did some work in this aspect, or like you have just collected the material?
My scholars are not Pradeep Kumar. He is working on therapeutic immunotherapeutics, so he has collected enormous data for designing a peptide for anti-COVID spikes, but he is not available today. That's why I have explained the properties of COVID viruses.
Yeah, so, it looks like a review kind of thing, no? That you have.
Yes, madam. And it is also more suitable for the title of the conference also. That's why I have prepared
this. But your presentation title, it should be like understanding the mechanism, not like that molecular basis of immune system. Instead, you should have put understanding the molecular basis.
Mechanism of the
that would have been better.
Yeah, it's a bit confusing only. Like it looks.
So you're just explaining the molecular basis.
Yeah, that's.
Yeah. Molecular basis for immune evasion mechanism, but IL-6 blocking also contributing for escaping from the attack of pathogenesis.
No,
so that's why doctors are prescribing montelukast and other anti-inflammatory drugs that are contributing more for the recovery of the patient.
generally the presentations are when you have done some, there should be some novelty in your work, no?
Okay.
Isn't it? When you are coming for such presentations, it should be something like, see, it is mere compilation of the data, isn't it?
Okay.
So you can mention like a review article, that kind of thing. Yeah.
So we have in first stage of designing therapeutics and searching for so many database. My scholars are working on this aspect in my laboratory.
Fine.
Thank you, madam.
Next participant.
Thank you, sir. Next participant, K. Usharani. I hope you are ready.
Yes, ma'am.
Please carry
Yes,
on.
ma'am. Yes, ma'am. 1 minute.
Please share your screen.
Yes, ma'am. I'm trying, ma'am. Is my screen is visible, ma'am?
Yes, it is. Please carry on.
Okay. Very good afternoon to all. Today, I'm K. Usharani, research scholar under supervision of Dr. Shanthi Burgula, Associate Professor and CBOS in the Department of Microbiology. And my title is Peptide Generated from Anchovies Using a Bacterial 16 Protease for Prevention of SARS-CoV-2. So proteases are the most significant industrial enzymes, accounts almost 60% of the global market. This bacillus species are most widely used in industrial enzymes. So in Japan, fermented soya product have been recommended as a therapy of SARS-CoV-2. The peptides identified from soybean fermented using bacillus species were showed good antiviral activity through in silico studies. What?
You're not audible, ma'am.
So in addition, several bioactive fermented soya.
Your voice
Hello?
is breaking. Yeah.
Activity against influenza virus, HSV, HIV, and a specific antiviral, specific. 1 second, I'm putting my earphones. Sorry. Coming to my present study, we used Bacillus subtilis from the soil microorganisms are able to generate peptides from the Stolopherus indicus, which is anchovies, as fish is a good source of biological active peptides in previous studies when used sardine. So we also tried to generate the peptides from anchovies. By using the bacillus cysteine protease. So peptides were analyzed through mass spectrometry, and these peptides were used for SARS-CoV virus entry prevention through in silico studies using GOLD software. So for screening, we isolated 5 organisms, and the organisms are submitted into NCBI, and we have done the protease assay, and among all A4 A4 isolate was showed a good taken this A4 for further studies. So we used Bacillus subtilis cysteine protease enzyme to generate the peptides, and these digested peptides were analyzed through mass spectrometry. So after mass spectrometry, we are able to generate peptides which are more in protein count. We have taken the further studies. So we have taken the peptides
And by using these peptides, we have done the ACE2 molecular docking studies. So we have taken the ACE2 structure from the peptide database, and by using this database, the peptides are able to bind to the active site of ACE2. So, and we got a good docking score. Simultaneously, we had tried with TMPRS to molecular docking studies using Gold software. So we have taken the structure and the active site of TMPRS SS2, and our peptides are able to bind to the active site of this TMPRS SS2, and we got a very good docking score. And the peptides also able to bind to the receptor binding domain of the SARS-CoV-2, and the docking score of peptides with ACE2, TMPRS2, and a spike glycoprotein given in the table form. So among all, peptide 10 was showing the good docking score, which is 74.99 for ACE2, and 99.1% for the TMPRSS2, and for spike glycoprotein, it is 66.86. So the conclusion of my work is the generated peptides upon LCMS revealed the presence of 3 peptides which are having the S2 inhibitory activity and TMPRS2 inhibitory activity. Peptide 1, 10, and 13 showed multiple sites of binding on the S2 enzyme active sites, mainly by hydrogen bonding. In addition, the TMPRS2 peptide 10 inhibitory complex hydrogen atom of peptide 10 formed 2 hydrogen bonds with oxygen atom of arginine 150 with a binding energy of 99.11 kilojoules per mole. So in addition, peptide 10 showed strong binding with the spike 2 protein in the receptor binding domain, forming a glycoprotein inhibitor complex by hydrogen bonding with nitrogen atom of lysine 343 at a binding energy of 66.86 kilojoule per mole. So interestingly, the binding of this peptide was observed to be away from the mutated location of the SARS-CoV-2 Omicron variant. Considering the multiple binding sites, these peptides can have a potential role in averting entry of SARS-CoV-2 virus in multiple ways. This could enhance results by prevention of binding of spike 2 glycoprotein to the ACE2 receptor on host cell, and also inhibiting the TMPRS2 mediated cleavage of virus entry. Thank you.
Nice. It's a good piece of work, actually. And like, what is the size of your peptide?
It's a decapeptide, and few are 12 amino acids length, ma'am.
Okay. Yeah. So how you can it is like how you can extrapolate your research further. It's a nice thing. See, if it is acting against different variants, so how you can go for further work on it?
Ma'am, we want to check this activity on cell lines and animal models also. So we are looking for that.
Okay.
You can present Vishali.
Yes. Am I audible? Sir, am I audible?
Yes.
Okay. Good afternoon, sir. Good afternoon, ma'am. My research topic is conceivable correlation amongst osmophilic bacteria B. satilis MZ315033 and leaven production along with the probiotic activity. Actually, this research work is divided into the 2 targets. First one, leaven mediated nanoparticle synthesis, and second one, we highlighted on the ability or the capability of the microorganism for the probiotic criteria. Now, all of us know that the osmophiles are nothing but which live in the high sugar environment, which have the ability to tolerate the highest sugar concentration. They are capable to grow in presence of the high osmotic pressure and yes, in presence of the low water activity. Now, levain is nothing but it is the polymer of the fructose. Yes, it is the polymer of the fructose. Now, next one, as concerned to the objectives, we want to isolate the osmophilic bacteria. Then the optimization was carried out by statistical method like Plackett-Berman design. Then we focused on the levain sucrase activity because levain sucrase have the ability to produce the levain. After that, we check the ability of the levain for the silver nanoparticle synthesis, and the last objective is study of the probiotic criteria of the osmophilic bacteria. Initially, we collected the different food samples which contain the lots of sugar. In that, we collected sugarcane juice, dates, honey, jaggery, sugar beet juice. You can see this table means naturally the sugars are present in these samples. Therefore, we selected these food samples. Next one, as concerned to the methodology, first of all, the sample collection, we collected the different food samples. Then the isolation of the osmophilic bacteria is carried out by using these foods. Then identification is based on the 16S rRNA sequencing. Yes, primary and secondary screening. Actually, we carried out the tolerance study. Means we increase the concentration of the sugars and we check the tolerance of the osmophiles, means up to what level actually they have the ability to tolerate the sugar concentration. Next one, we check the ability of the microorganism for the levain production. Then the characterization is carried out by the TLC, by the HPLC. And the optimization study was carried out by the statistical method like Plackett-Berman design. And yes, levain sucrase production was also carried out by the selected microorganism, and we checked the presence of the levain sucrase enzyme by the SDS page electrophoresis. Next one, levain mediated silver nanoparticle synthesis was carried out, and we checked the probiotic criteria of the microorganism, means Bacillus satilis. Okay, these are the results. You can see this is the screening. During screening, we observed that these three bacterial strains showed the ability to tolerate the maximum sugar concentration, means up to the 100% sugar is tolerated by these microorganisms. And yes, in presence of such a high concentration, they have the ability to show their growth on the surface of the nutrient agar plates, which contain the maximum sugar. Now here, yes, there is the one organism S1 which have the ability to tolerate the maximum sugar concentration. Yes, now here you can see this nutrient agar plate, means the normal sucrose is present, normal in the sense 3% is present, and here we are taking the 100% of the sucrose, and you can see the difference in between the both, because in presence of the high sucrose, in presence of the high sugar, some changes are occurred as concerned to the morphology of the bacteria. We can see the mucoid colonies. Okay, next one. This S1 isolate we selected on the basis of the high sucrose concentration tolerance. Okay, and simultaneously it have the ability to produce the levain also. The gram staining is carried out, it is gram positive, and yes, it is the capsulated microorganism. Then on the basis of these results, the molecular identification is carried out. Now the sequence data is get totally matched with the Bacillus satilis. Okay, then on the basis of the similarity, phylogenetic, on the basis of the similarity of the phylogenetic analysis, yes, this organism is the Bacillus satilis, and we submitted this sequence data in the NCBR. Now, next one, we also studied the total soluble sugar. Okay, total soluble sugar which is present in this food sample. Now, next one, we estimated the reducing sugar. Actually, when this Bacillus satilis is responsible to utilize the carbon source. Okay, means it have the ability to utilize the sucrose, which is a ready-made carbon source, but instead of a sucrose, we use the jaggery. These organisms also have the ability to utilize the sugar which is present inside the jaggery as a carbon source. And yes, here you can see the reducing sugar is increased up to the 48 hour. This is the estimation of reducing sugar. Yes, now this is the TLC. Now, this leaven is nothing but it is the polymer. It is the polymer of the fructose. The spots which are present at the bottom, these are actually the spots of the leaven. And at the top, we can see the spots of the glucose. Yes, this is the HPLC analysis. And for that purpose, we taken the standard sucrose. This is the optimization study. We already taken the 7 parameters and depend on the p-value. The p-value is less than 0.05. Therefore, according to this analysis, the sucrose is the significant factor which affect on the production of the leaven. Yes, this is leaven sucrase activity which is increased at the 48 hour in presence of the sucrose and in presence of the jaggery also. Yes, this is the SDS page electrophoresis. You can see here the molecular weight of this enzyme is up to the 50 kD. And yes, this is the band. It shows the presence of this leaven sucrase. Yes, we separate the enzyme, we purify the enzyme, then the SDS page electrophoresis was carried out. Yes,
Vishali,
yes, yes.
Okay.
Okay. This is the nanoparticle synthesis. Yes, now this is the conclusion. These organisms have the ability to produce the leaven mediated nanoparticle synthesis, and yes, on the basis of antibiotic susceptibility, antimicrobial activity, and hemolysis test and the protease activity, we can say that this organism is significant to fulfill the criteria of the probiotics. Therefore, the whole method is versatile one. It is eco-friendly one. This is the conclusion. Thank you.
Yeah, hello. Yeah, Dr.
Yes, sir.
Vishali. Yeah, did you purify the enzyme?
Yes, sir.
Did you purify the enzyme?
Yes, sir.
Yeah, you have shown a single band, but you didn't. Yeah, which method of purification have you used?
Sir, actually, initially we used ammonium sulfate purification method.
Did you get a completely purified a single band with ammonium sulfate purification?
Sir, actually, after ammonium sulfate purification, we perform the dialysis method also.
Yeah, but still, I don't think you can purify enzyme with ammonium sulfate.
Even I would like to ask
and sir, that particular sample actually we run by using the column chromatography. I used the silica gel column, and I collected the filtrate.
Yeah, but anyway, okay. But I am just wondering, did you get a single band with that 2 steps or 3 steps?
Yes. Sir.
Are you sure?
After 3 steps, sir.
Okay.
Actually, ammonium sulfate but precipitation, then dialysis, and then the column chromatography.
Yeah, but still, it is not so easy with. I mean, it doesn't have. You don't get a single band like that.
Yes, sir.
Okay.
Actually, anyway, we faced lots of difficulties.
I think it is. I don't think it is completely purified. It's a partially purified enzyme, and you have done some work on it. Anyway, okay. Over to others.
Dr. Vishali, did you try RSM for this optimization?
Yes, ma'am. Actually, we will go for the response surface methodology, CCD.
Yeah, you tried?
Yes. No, ma'am. That work is actually remained.
No.
Yes, this is plaquette Berman. Ma'am, actually, plaquette Berman is only responsible to show the significant factors. But we should go for the RSM because it will show the specific values.
That's what.
Yes, ma'am, definitely we will go for it.
Yeah, and one more thing. How about the economics involved in that, like commercialization? If you see the commercial aspects.
Yes, ma'am. Actually, economical in the sense, osmophiles have the ability to utilize the sugar which is present in the jaggery.
That's fine.
Yes, it is.
If you compare it with other organisms, like how your organism is better if you see the total processing cost and all.
Yes.
So yes, you feel that,
ma'am, actually comparatively the jaggery is economical, means as compared to the
you can even try some more substrates also, isn't it?
Yes, ma'am. Ma'am, actually I tried the number of substrates, sugarcane juice and all, beet juice. Yes, dates we also tried, ma'am. But the better results we observed in this particular jaggery. Okay. And these organisms, ma'am, actually responsible to produce the leaven in a large amount compared to the other, compared to the other substrates.
Yeah, then you can see like after optimizing the conditions, how you can improve it further. That you can
Yeah, definitely.
go for RSM and all. Yeah.
Yeah,
Anyway,
definitely, ma'am.
Okay,
Yes.
fine.
Thank you. Thank you very much.
Welcome, Lakshmi ma'am. Welcome to this session.
Hi, sir. Is my screen is visible now?
Yes.
Thank you, Dr. Vishali. Pavan Kumar, please
Yeah.
continue.
Yes, ma'am. Good afternoon, all. Thank you for giving this great opportunity to introduce myself. This is Pavan Kumar, research scholar working as SRF under the guidance of Professor Smita C. Pava at Usmania University. Here now I'm going to give a seminar on bioinformatic analysis of system-derived dysregulated genes from COVID-19 patients targeting the C gas and sting pathway. The coronaviruses are a group of enveloped and single-stranded RNA viruses ranging from 60 nanometer to 140 nanometer in diameter and 26 to 32 kilobase in length.
Pavan, go for full screen. Pavan, go for full screen.
Sorry.
You can go for slideshow.
Pavan, also, please, can you skip about coronavirus? Everyone knows what is the size and infection. Talk about your work, no? That will be better.
Yeah, sure, ma'am. Skipping now, okay, ma'am.
Yeah.
Yeah, sure, ma'am. This mainly the structural and phylogenetically identical to source COVID and is made up of 4 major structural proteins: spike, envelope, and glycoprotein and nucleocapsid.
Your slides
protein.
are not moving, Pavan. Yeah.
Is it not moving, sir?
No, now fine.
Okay,
Yes.
sir. And this due to the mutations is the spike protein regions, there are so many 4 types of new variants are releasing. Those are alpha, beta, and delta, and gamma, and Omicron. The recent beta 1.152 and Omicron variant spike releasing. Among the most concerning receptor binding domain mutations found in this Omicron are E484A and N501Y and S417 and so on. All of these have been previously observed in major variants of concern like alpha and delta. When SARS-CoV enters into the body, the inflammatory response acts as an antiviral defense. But a severe cytokine storm caused by an imbalanced reaction can be extremely harmful to the patients. As a result, utilizing ways to successfully suppress the cytokine storm. When the SARS-CoV enters into the human body, it causes a cytokine storm, means alter the cytokine expression. So, for example, these interleukins and interferons. Due to the cytokine storm, maybe various bacterial infection and fungal infections arises, and also tissue damage also arises. After this, various body parts are going to damage, example, lungs and gastrointestinal tract and brain and cardiovascular, liver, kidney, eyes, and these are all here illness and symptoms arising illustrated in here. In the context of infection, the cellular stress and tissue injury, the C-gas sting signaling pathway has emerged as a critical mediator of inflammation. The ability of the C-gas sting pathways to monitor and control the cellular response to microbial and host-derived DNAs, which serve as ubiquitous inflammatory associated molecules. The C-gas sting pathway, C-gas, cyclic GMP and AMP synthase is an essential molecule for controlling the baseline expression levels of cell-intrinsic immune genes in cells. Making here interferons and interleukins induces OAS, oligoanalase synthase enzyme. When induces this OAS activates the CGMP, and activation CGMP will activate sting, and from the sting TBK and NFKB pathways are arising. From this NFKB, the various alternate pathways and degradation of viral RNAs. And here another pathway from this OAS activates RNase L. By the activation of RNase L, it degrades the viral RNAs and also viral DNAs at cellular level. The IFN pathways are directly engaged in virus spread protection and are critical for the initial cell intrinsic antiviral response. The inflammatory cytokine response is important in immune cell recruitment and activation. So here, interferons and interleukins play an essential role in the pathogenesis of COVID-19. In our study, we performed the analysis of blood PBMCs and inflammatory transcriptome of COVID-19 datasets from GEO. This is the schematic approach of DEG's validation, comparative data analysis, Venn diagram, KGGF means enrichment, PPA interactions, and selection of DEGs using of a cytohuba. Our methodology includes data analysis by GO2R and a lemma package of R with using of Benjamin Hedgehog false discovery rate statistics with utilizing log fold change 0.5 and p-value less than 0.5 criteria. And DG's analysis were carried out using DSEC and common genes identification between two datasets and enrichment analysis of DG by enricher and protein-protein interaction network were constructed using the string database and identification of hub genes by cytochrome plugins. And prediction of these drug candidates were identified hub genes by drug signature database and docking and dynamic simulation analysis of these predicted drugs by using MVD and PyRX and MD simulations using by GROMAX and also CHARM. Our result is a dataset normalization for identification. The volcano plots here states that the down like blue color and red color states about the down and up regulation of DEGs. From this every dataset we have identified a total 599 means from this dataset we identified total 599 DEGs, whereas 91 upregulated and 4 downregulated DEGs are identified. From this dataset we have identified a total 784 DEGs in which 107 upregulated and 38 downregulated DEGs. Among these two data sets, the common DGs, common dysregulated genes are 296 we have identified from this. To facilitate the visualization of the top 100 DGs, we aggregated the heat map and their p-values. Here we mention this upregulated DGs from the two data sets and also downregulated DGs from two data sets. These are the common DGs of 296 of these both data sets. The gene ontology and pathway enrichment analysis were carried out by the enricher. Here, most of the DEGs were involved in cytokine-mediated signaling pathway. Most of the upregulated DEGs biological function is cytokine-mediated signaling pathway, and this molecular function is a cytokine-mediated receptor activity, and the cellular component is upregulated DEGs were involved in integral components of the plasma membrane and secretory granule membranes. Among these 198 upregulated DGs, 69 genes were involved in the cytokine-mediated signaling pathways, 49 were cellular, and 36 genes were inflammatory response. And this is downregulated genes. These are also involved in the same process. And here, among these 42 downregulated genes, 7 genes were involved in the cytokine-mediated, 15 were involved in cellular response, and 10 genes of downregulated genes were involved in inflammatory response. These are the wiki pathways. Wiki pathways, KEGZ, reactome, and biocarta pathways are analyzed here. After analyzing, these are all included in the cytokine-mediated string responses. This is the PPA interactions. After PPA interactions of these 296 common DEGs, we have identified the top 10 DEGs from the cytohuba, and top 10 DEGs significant values from these both data sets are supporting to our study. And brief introduction about the top 10 DEGs what we have identified. These are also having the role in COVID-19 prognosis of poor survival. And this is drug prediction. After identifying top 10 genes, we are just docking with this top 10 genes with using of COVID-19 main protease enzyme. After that, we didn't.
Pavan, your slides don't seem to move.
Yeah, now moving.
Please conclude, Pavan Kumar. Please.
Yeah,
conclude.
sure, ma'am. Yes, our molecular studies and dynamic simulations were concluded that digoxin is a main ligand, can inhibit this main protease and reduces the cytokine storm in COVID-19 patients to reduce this symptoms of COVID patients at the lung and blood cells. Our conclusion, this work has provided the groundwork for identifying prospective candidate gene biomarkers, combating gene RNA network, and sterilizing drug repurposing mechanisms to treat COVID-19. As a result, it is significant that this provides comprehensive answers to combat the pandemic by revolutionizing novel diagnostic and treatment methods. These are the general references which supports to our study. And thank you.
So Pavan, like your sample size is quite small, isn't it?
Yeah, actually, ma'am, less samples. These two data sets are recently uploaded in GEO. One data set is inflammatory data sets for skin biopsies, and another data set is PBMCs, peripheral blood mononuclear blood cells of blood samples. We have analyzed these two.
Yeah, because you have taken it is very less, isn't it? Like what is basically, you should go for a little more, isn't it? The sample size should be more, no?
Yeah, yes, ma'am. Actually, the samples in here, we included total six control samples and 15 COVID-19 patient samples, ma'am. And we removed, yes, ma'am.
Yeah, this is too small, no sample size for a study.
Yes, ma'am. Very rare data sets are there, because lung, other data sets are also there, but already they reported. But these two data sets are
And
only.
this is for which this thing variant? For which variant you have taken from Beta or?
Actually, in those papers, they included that some are Delta and very less Omicron, ma'am. Recently uploaded data.
Okay, but it is like it is quite different, no?
Yes, ma'am. Actually,
Pathogenesis and.
Yes, ma'am. What we have identified in previous studies, they identified IL-6, interleukin-6, and IL-1 beta is a prognostic biomarker. In our study, we have identified not only those interleukin-10, also a prognostic biomarker.
So, Pawan,
Ma'am.
so have you noted the age and sex of the patients and any comorbidities in the patient?
Sorry, ma'am.
Age and sex of the patient.
No, ma'am. We didn't include those.
What is their comorbidity? You know, out of the 4 patients, were some already suffering from some other ailment like diabetes or?
Yes, ma'am. From those data, some people are so it means illness with cancer, but we are excluded those samples, ma'am. We are just included only COVID-19 positive patients only. We are just included some cancer patients, and also we are excluded some skin samples who is suffering with the CLS. We are including only the controls with COVID-19 positive patients.
What about thyroid disorder, diabetes, and hypertension? You did not exclude those, right?
Yes, ma'am. We excluded all those things, ma'am.
So that means the patients are only suffering with
COVID positive.
COVID, but they were all healthy.
Yes,
Did you also have that information that they don't have any other ailments?
No, ma'am. Actually, we didn't.
That's what, you know, because that will also make a lot of difference. You know, the cytokine profile is different when there is hypertension. We don't know what is the state of the patient, what is the status of their actual ailment, you know, like diabetes. Comorbidities are there in COVID-19 patients. So maybe you have to first classify them, you know, according to their comorbidity plus their age and sex, and also what is the severity of infection. That's what I think.
Yeah, yes, ma'am. Sure, ma'am. And actually, in previous studies, we have done this interaction with COVID-19 patients with mucormycosis, with the sex and age, and who is having illness with the diabetes and who is suffering with the cancer. But in this study, we just excluded these all things. We just included only PBMC of blood samples with skin biopsies only, ma'am.
Okay.
Thank you.
Thank you, Pawan Kumar. All the very best. Next, I invite Nichita to please present your screen. Nichita ma'am, can you unmute yourself?
Let's see, call next person.
Looks like she's having net problem. Maybe you can ask her to present later.
Yeah, next two you can call.
Okay, ma'am. K. Swarnalata, if you're ready, can you please present your presentation? K. Swarnalata. Dr. S. Sai Bhavani.
Swarnalata, given co-host. You can start.
Swarnalata, can you please unmute and start your presentation, please?
Excuse me, ma'am, can you make PL Shravani as co-host as I'm presenting from the laptop?
Yeah, sure. Just a minute.
I have made it.
No.
Yeah. Who is presenting?
Shravani.
Okay, Shravani. Okay, 1 second. PL Shravani. Okay.
Can you help me?
Sir, I think Swarnalata
Swarnalata is presenting from Shravani, sir.
Shravani.
Swarnalata
Okay,
is the
okay.
participant.
Fine, fine. Thank you. Okay.
Nishita, you just wait. Yeah, next you can present.
Sir, can you see my screen, sir?
Yes, now it is come.
Is it visible, sir?
Yes,
Yes.
please carry on.
Okay. So good afternoon, everybody. I, K. Swarnalata from Department of Microbiology, St. Pius X Degree and PG College for Women. My topic for today is COVID-19 safety, awareness, and adverse effect of vaccines. It's a review article. So quickly, I would just brush up what is COVID-19. As we all know that COVID-19 is caused by SARS-CoV-2 virus. So it was first observed in China, and we are aware of how is it transmitted by direct and indirect routes and by respiratory secretions and the associated symptoms. So I would like to concentrate more on the variants. So there are emergence of new variants like alpha, beta, gamma, delta, Omicron. These are considered as variants of concern because they are showing high rate of transmission and severity. So the question is, are we at more risk, and will the same vaccine be helpful or medication be helpful? So how to protect from getting infected is, first of all, the first line people who are working must be protected by using the specialized shields like PPE kits and all the social measures like wearing a mask and social distancing. All these have to be maintained. So though there are several drugs which are available which act as anti-SARS-CoV-2 such as remdesivir, then favipiravir, then azithromycin, it has shown a remarkable result for the patients to recover from severe sickness. But taking a vaccine is a must. So these medicines usually complement the vaccine, but actually they don't replace as such the vaccine as such. So it will also only reduce the burden which is associated with the infection, that is getting hospitalized, or people getting vulnerable, like who cannot be vaccinated or whose immune system is low. So a safe and effective vaccine, when taken, it acts as a game-changing tool. So how COVID vaccine helps us is nothing but, as we all know, that vaccine will train our immune system so it can protect us and our community. It can help slow the spread of infection also. So every individual must be vaccinated. The reason is, when most of the people in a community are protected by vaccination, the ability of a pathogen to spread will be limited. It will help in developing herd or indirect or population immunity, generally called as herd immunity. So when people have more people are immunized, it will help us protect, and it will help people who cannot be vaccinated, like immunocompromised people and people who cannot be vaccinated. So in order to ensure the safety of the vaccine, the introduction to vaccine to the public involves various steps. It includes vaccine development too. So many different public organizations and many clinical trials and FDA authorizations and manufacturing and distributions have come up. And these public and private companies all over the world, they have come up with a suitable vaccine, and it is made available to the public, like Pfizer, Moderna, Johnson and Johnson. These companies, they have come up with different kinds of vaccines. All these steps will usually help in ensuring safe and effective vaccine. So though vaccines are available to the public, still we have not achieved 100% vaccinated population. The reason is still hesitancy exists among the population.
this is because people have fears like vaccines have side effects, they have active viruses within them, and people who are pregnant do not take vaccine, and women who are under menstrual cycle, they don't take vaccine. So all these are leading obstacles to achieve 100% vaccinated population. And it has been observed that though people have taken vaccine, still hospitalization and death cases have been reported. The reason is, again, still some people are not vaccinated, or there is emergence of more transmissible variants among the people, and the relaxation of social measures of not wearing mask, or the underlying health conditions contribute to the fear among the people. So as though we know that as a microbiologist, we say that vaccines are effective and it will protect, but every vaccine some or the other way have got side effects. These side effects can be very mild, like having fever, fatigue, which actually, if taken in a positive sense, it is actually boosting the immune system. It is triggering our immune system and telling us that, yes, so it is working well. So apart from the normal side effects, adverse effects are observed. Like some people have undergone, after vaccination, they have taken up, they have undergone heart problems, and some people have developed Guillain-Barré syndrome, and some associated symptoms like headache and blurred vision. So with this, I would like to say that vaccines are available and it should be taken. Everybody should take the vaccine dose, and the dosage should be completed to control the spread of infection. And a structured awareness among people of whether rural or urban areas, and continuous training and education to public must be provided for safety and efficacy of the vaccine. And the Ministry of Healthcare and WHO also recommends that everybody should take vaccine for a safer life. I would like to conclude, and I would like to thank the organization, Palamur University, and the Department of Microbiology for giving me this opportunity. Thank you.
Thank you, Swarnalata. All the very best. The next participant, Nichita. Ma'am, can you please start sharing your screen?
Good afternoon, everybody. I am Nichita, lecturer in microbiology in TSWRDC Suryapet, and student Dr.
Nichita, your voice is not clear. Your voice is breaking.
You do one thing, switch off your video and present oral presentation. You explain it orally. Switch off your video and you can explain your content.
Okay, sir. I will do it. Okay, fine. So, can you see my screen, sir?
Yes, ma'am, continue.
Yeah. Okay. So sepsis is a condition where immune system starts affecting its own body, leading to organ dysfunction or to extent death of an individual upon bacterial infection. The main reason behind this cause could be due to unregulated and uncontrolled production of cytokines. So in our laboratory, we try to mimic the bacterial infection in whole blood culture with the lipopolysaccharide extracts, and found that only 10 nanograms of LPS was sufficient to initiate cytokine storm. And they were very crucial peptides required for cell survival and also protecting cells undergoing apoptosis. And it was found after extraction of mRNA from LPS blood culture by Trizol method and then subjecting it to qRT-PCR. The time points maintained were for 6, 12, and 24 hours. The mRNA expression was confirmed by running SDS page and also Western blotting of serum sample after acetone precipitation. And to understand the role of therapeutic intervention to save a cell undergoing apoptosis, retinol in form of palmate was administered in 5 nanograms concentration, as it has an anti-inflammatory effect and also implicated in the cell survivability. So the results which we have got is, upon LPS induction, anti-inflammatory cytokines mRNA expression was upregulated to 100-folds. Cytokines like IL-6, TNF-alpha, TLR4 were shown upregulated until 24 hours. And cells which received retinol in form of palmate could overcome from septic shock and caused by LPS and protected themselves undergoing apoptosis. After addition of retinol, cells started showing downregulation of cytokines and could stop the cytokine storm, which is shown in the Western blot analysis and also RT-PCR. So I would like to conclude that only 10 nanograms of LPS was sufficient to induce blood a 100-fold increase in the cytokines and initiate cytokine storm and leading to the death of the cells, which was confirmed by flow cytometry, where cells induced with LPS had an mean fluorescence index of 14.7, where untreated cells showed a mean fluorescence index of only 6.74. When retinol was added, the mean fluorescence was decreased and the cells did not undergo apoptosis. The cells which were not given a therapeutic approach of retinol have undergone apoptosis, indicating that bacterial sepsis can be a life-threatening condition, silently and very profoundly initiating cytokine release, which later converts into a cascade of unregulated cytokine, leading to organ dysfunction and hence death. If a person undergoing sepsis is only treated with antibiotics, could not be saved. He has to be also treated with other anti-inflammatory injections so that bacterial infection and simultaneously the cytokine storm which is generated will be stopped and the person will be survived. Thank you.
Questions?
Yeah, you have used the RT-PCR to identify the interleukins.
Yes, ma'am.
So before that, you ran on the gel, the interleukins.
Yes, ma'am. Yes.
So the primer designing and all has been done by you.
Yes, ma'am. We have only done it.
So you have taken only the blood cells as the model system, and then you are applying even to the whole organism. You would like to take it as a model system.
No, ma'am. Till now we have selected only whole blood culture, ma'am.
So you want
in
to
our
apply
lab,
to
other things are also going on. Yes.
No, you want to apply to the higher order model systems, just apart from the blood culture.
Yes, ma'am. We have done it on the cell lines also.
Mm-hmm.
But it is not presented here. But we have done it.
You could get the same results.
Yes, ma'am. The results were same.
With the lipopolysaccharides of the same concentration could elevate the cytokine
Yes,
storm.
yes, ma'am.
To the same extent, even
Yes,
in the
ma'am.
cell lines.
To the same extent. Yeah, even in the cell lines.
Okay. You are
So
having
which cell lines did you use? I'm just intervening in between. Which cell lines did you use, Nichita?
PHP1 cell lines, ma'am. And also A549 cell lines, ma'am.
Okay, because see the condition when you are going only for the conditions, no, like only blood, it will be quite different because there are many other factors which are also involved when you are going for a model system or cell lines, no? So I think you have to consider those aspects as well.
Okay.
Thank you, Nichita ma'am. All the best.
Thank you, ma'am. Thank you so much for giving this opportunity. Thank you.
Next, I invite Swati.
Okay.
Please present your presentation.
Yes,
Carry
ma'am.
on.
Good afternoon, everyone. I'll quickly introduce myself. I'm Swati Bantu. I'm pursuing my PhD under the supervision of Dr. G. Shamprasad sir, Department of Microbiology, Chaitanya Dimuthubi University, Warangal. And today's my topic of discussion is potential of vegetable waste to synthesize metal nanoparticles for nanomedicine formulation. Coming to my topic, there are a number of studies that has been shown that there is an increase in prevalence of invasive fungal infections, especially in immunocompromised patients and such as HIV and diabetes, and also patients who has been hospitalized with serious underlying diseases. So Candida species mainly belong to normal flora of oral cavity, gastrointestinal tract, and genitourinary tract, which may be responsible for some serious clinical infections. Coming to Candida species, Candida infections, these are of two types. One is invasive type of Candida infection, and another one is superficial kind of Candida infections. Here you can see in the image you can see that there are different parts of body where Candida infections can be occurred, like mouth, skin, nails, vaginal candidiasis, and pineal. And also these Candida can invade into organs such as eyes, brain, bones, lungs, and heart. These are some images showing superficial Candida infections on skin and oral cavity of patients. So development of drug resistance by fungi. In recent years, there is a huge record of resistant fungal agents that that has been recorded. So why these fungus are gaining resistance against antifungal drugs? This is because they are exposed to continuous increase in the antifungal drugs. So these increase in the resistance strain necessitate for search for new antifungal agent. In this aspect, nanotechnology, which mainly involves in the nanoparticle synthesis, is giving a scope for creating new therapeutic agent, which effectively acts against resistant fungal strains with different microbial targets. Coming to toxicity of chemically synthesized nanoparticles. Chemically synthesized nanoparticles with various concentrations are proven to be carcinogenic for human use, as well as these are toxic for environment also. So the toxicity of nanoparticles generally leads to generation of reactive oxygen species, oxidative stress, inflammation, etc. So the biological methods are mainly proven to be effective for humans and as well as environment. So usually chemical processes require some toxic chemicals such as hydrogen boride and sodium borohydride, which are very toxic for human use as well as environment. So in the green chemistry or green synthesis of nanoparticles, we are using eco-friendly reducing agents which are acquired from plant basis and also microorganism basis. So here in this context, we are only focused on plant-based nanoparticle synthesis. In this present picture, you can see that process of nanoparticle synthesis mainly involves two steps, that is reduction of metal precursor to metal ions and the stabilization of metal ions to metal nanoparticles. So this reduction and stabilization is involved with some reducing agents and also stabilizing agents in chemical synthesis, which are very toxic chemicals has been used. Coming to green synthesis, the phytochemicals that are present in vegetables, plants, and different extracts of a plant are used as reducing as well as stabilizing agents. As we can see from the Food and Agriculture Organization, FAO, it has been claimed that India stands second in the production of vegetables, but almost 40% of produced vegetables have been wasted in the process of harvesting and harvesting to reselling. So the essential nutrients which are present and phytochemicals which are present in these vegetable waste can be used sustainably in the synthesis of nanoparticles. So the green synthesis of nanoparticles has main advantages such as these are economically viable, environment friendly, enhances efficacy, improves the reactivity, and also the main concern is it reduces the toxicity. These are some images of vegetable waste. So the main objectives of my work is to synthesize mono, bi, and trimetallic nanoparticles using silver, copper, gold, and platinum with the different vegetable waste concentrations, and to characterize the synthesized nanoparticles, and also to identify antifungal activity of different formed nanoparticles upon different fungal species. And the final thing is to formulate a nanomedicine ointment to act effectively against resistant isolates of Candida species. Till now, I'm at preliminary stage of my work. So I have synthesized silver nanoparticles using spin guard and bitter guard. I have collected some spin guard from a local market.
Please
and
conclude. Swati, please conclude.
Okay, ma'am. So the main aim is to synthesize bi and trimetallic nanoparticles, where compared with monometallic nanoparticles, bi and trimetallic nanoparticles have a wide range of properties. So I am focusing on synthesizing tri and bimetallic nanoparticles using this vegetable waste, and also to formulate that nanomedicine which effectively acts against upon resistant strains of Candida species. Thank you, ma'am.
Yeah, Swati.
Yes, sir.
Do you have any reference which says that chemically synthesized nanoparticles are carcinogenic compared to green synthesized nanoparticles?
Sir, it is actually in chemical synthesis of nanoparticle, we use different kind of chemicals which are very costly, and also they can harm environment as well as human body. And I have some references, sir.
No, so you mean to say those solvents or chemicals enter the human body after synthesis?
Sir, after consumption or
for synthesis they are required.
Sir?
They are required for synthesis. They don't enter the human body. Do they enter the human body?
Sir, for nanoparticle synthesis, they will for stabilization of nanoparticle, this chemical can fabricate the nanoparticles, sir. That might affect the human body, which we take as a medicine. If we take it as a medicine.
No, ma. That concept is wrong. Whether it is vegetable waste, biologically synthesized, or chemically synthesized, or the physical methods which are used, they don't the effects are same. Okay, there may be different, as you said, that there could be differences in whether it could be economical or eco-friendly, or these are different. But it's not that no, if they are carcinogenic, even these also will be carcinogenic. Biological synthesis.
Yes, sir.
Okay, fine.
Thank you, sir.
Actually, Swati, your title says about synthesis as well as about its antifungal properties, no?
Yes, ma'am.
But see, till now you have just synthesized, I believe, isn't it?
Yes, ma'am.
But then you can't give the title this thing, no? Because that is your future prospect.
Yes, ma'am. I have to work on it, ma'am.
Yeah, but because when you are presenting,
okay,
it's not, you have not done the work, isn't it?
Yes, ma'am. Yes, ma'am. I have just started my work.
Yeah, so I think it is like it's a little misnomer, like what you are presenting. It's okay. Synthesis is fine.
Okay.
But talking about its antifungal properties and all, no, unless you have finished that work.
Okay, ma'am.
Can't give like that.
Okay, ma'am.
Okay.
I'll consider.
Next, Priya's Ravan.
Thank you, Swati. All the very
Thank you,
best.
ma'am. Thank you, ma'am.
PL Shravani, can you please?
Yes, ma'am. Very good afternoon. Yeah. Ma'am, hope my screen is visible.
Yes.
Yes, a very good afternoon to one and all present. So I'm PL Shravani from the Department of Microbiology, St. Piston Degree and PG College for Women. So the topic is the alternate weapons for treating the COVID-19 pandemic, which is a review article based on the literature. So coming to the introduction, we all know that
We lost your presentation.
Please unmute Shravani. Try to share your screen again.
second one is the dalbavancin, which is used to treat skin and the skin-related infections, and the third one is the glycyrrhizine or the licorice root.
Shravani, 1 minute. Your screen has not shared. Share once again.
1 second, sir. I'm sorry. Sir. Sir, now, sir?
Yes, it's fine.
Yes. Is it okay, sir?
Yes.
Yeah. So the third one is the glycerine or the licorice root, which is used in the traditional Chinese medicine. And the fourth one is the recent drugs, which were recommended by WHO recently in the year 2022 in January, that is on 22nd. The first one, which is the teicoplanin, which was first identified in India and from the organism Actinoplanes teichomycetes, and it is sold under the brand name Targoxit. And coming to its structure, it is basically non-ribosomal heptapeptide with 7 aromatic amino acid chains and with the sugar residue, as well as a lipid chain, whereas the lipid chain acts as the hydrophobic tail, and as the rest part acts as the lipophilic moiety of the teicoplanin. So basically it is used to treat, and this is the diagram which enhances or which summarizes the teicoplanin mode of action in the gram-positive bacteria, where basically the lipid tail comes and binds to the L-alanine and L-alanine, the subunits or the precursors which are present in the peptidoglycan layer of the gram-positive cell wall, thereby inhibiting the polymerization and the cross-linking steps in the cell wall synthesis in the gram-positive bacteria. And so our concern is, how is teicoplanin inhibiting or inhibiting the virus replication? So it is 2 different mechanisms have been proposed by teicoplanin to inhibit the virus replication in the host body. The first mechanism is the inhibition of the cathepsin L protease, and the second mechanism is by the inhibition of the cysteine protease. So coming to this is the clear picture which shows the 2 different mechanisms which have been proposed by the teicoplanin, which shows the inhibition of the viral replication. The first one is by the cathepsin L activity. So this cathepsin L is basically a protease member which is found in almost all the cells and all the tissues of the human body. The main function of this cathepsin L protease is to recircle or recycle the proteins which are generated by the antigen pathogen endocytosis, and its concentration also increases in the chronic inflammation. So what is this teicoplanin coming and doing? This teicoplanin coming and it's binding to cathepsin L, and this cathepsin L is required for the interaction of the spike protein and the AC2 receptor for the release of the viral particle into the cytoplasm. So what it is doing, this teicoplanin is coming and binding to this cathepsin L protease, and it is inhibiting the release of the virus from the late endosome into the viral, that is into the cytoplasm. And the second proposed mechanism is blocking this 3CL Pro activity. This 3CL Pro is the major protease which is found in the coronavirus and is a highly regulated, and it is present at the 3' end, and its main function is to break down the complete protein into 11 different functional proteins, so that which is required for the virus assembly. Again, this teicoplanin is coming and blocking this 3CL Pro activity, which is inhibiting the virus multiplication. So in this way, the teicoplanin is inhibiting the virus entry, viral multiplication, and inhibiting the virus. And the second one is the dalbavancin, which is a novel semi-synthetic antibiotic where most of the countries have been started using this drug from the November 2021, and it is a semi-synthetic lipoglycopeptide antibiotic. And the best advantage, and it is the highest treated target of the drug, because it has the capacity to increase its half-life. So almost 300 to 310 hours it has an increased half-life, because it binds to the blood serum proteins. So the administration of this dalbavancin in very small concentrations also is very enough to treat and it is a very potential target for treating the COVID-19 infection. And coming to the third one, which is the glycyrrhizine or the licorice root, which is used in the traditional Chinese medicine. And 85 to 90% of the corona infections have been in China have been treated with the GA or the licorice root, that is the traditional Chinese medicine, because it is actually the plant actually is an origin of Mediterranean Sea, but now we have also started growing in India and Russia because of its advantages. And the main advantage is because of the phytochemicals, that is the root has phytochemicals, which are these chalcones and triterpenoids. These chalcones are the organic compounds which have the major advantages like anti-inflammatory, anti-cancer, and it has helped in treating diabetes. It has n different number of advantages, and this GA is also used in treatment of HIV, herpes simplex virus, and even in corona. So these are the 4 different ways in which this GA is inhibiting the activity of coronavirus. The first one is by proliferating the T cells, because the virus, most of the COVID patients have decreased T cell because the virus keeps on evading the immune evasion and starts decreasing the number of T cells. And the second one is the GHGMB1, which is the morbidity group complex box, which is present, and it is present in our tissue cells and is associated during high chronic infections. And this GA will come and bind to this and stop the multiplication. And the third one is the domain of lipid rafts. This lipid rafts is nothing but this is present on the surface of all the cells and is associated with the cholesterol. So that is the reason diabetic patients are more prone to coronavirus, and even their prognosis also becomes late because of this the lipid rafts. This GA pulls down the association of this lipid rafts and reduces the cholesterol level. And in another way, it is also
please conclude.
Shravani,
ma'am, please conclude.
Yes, ma'am. And it is also treating the diabetes also. And these are the recent drugs which have been proposed by the WHO recently in January 2022. And this is the conclusion. So all these different drugs treat COVID as well as bacterial co-infections in a very drastic way. And most of the drugs have been used from 2021 August. And still, even we are doing a research, we are trying to get different samples from hospitals so that we can put it onto paper. Thank you. And I thank the organizers for giving me the opportunity.
Shravani, are you working in this area?
No, ma'am. Actually, we are thinking of doing it, ma'am. Actually, we have done the research.
This is just a review, no?
Yeah. This I have mentioned, ma'am. I have mentioned it.
Yeah. So, are these medicines available in the market?
Yes, ma'am. Definitely, ma'am. Most of the countries have even started using these drugs. There is a very well statistical analysis given by most of the research work, and most of the countries have started using these drugs also. Along with, they also treat this resistant gram-positive infections also, which are caused other to the COVID virus infection.
Yeah, are you sure? Are you sure these drugs
Yes,
are in
yes,
market?
sir. I'll send you the reference links also, sir. I am pretty sure, sir.
Which country these drugs are being used, ma'am?
Sir, it is already using. They have started using in Canada and UK. In India, no, sir.
In Canada and UK, they are using this drug.
Yes, sir. I'll send you even the reference links, sir.
Fine. Okay.
Yeah.
Thank you,
Okay,
Shravani.
sir.
All the very best.
Yeah, thank you.
Next, I invite
Pratibha.
Okay. Pratibha, to please share your screen, please.
Good afternoon, all. I am Pratibha from Microbiology Department, Aluminium. Okay. Yeah. So I'm am I audible? Hello? Hello?
Yes, ma'am. Yes, ma'am.
Yeah. So my topic is NMOX for wastewater treatment. It's a NMOX, the full form is an anaerobic ammonia oxidation. Yeah. Anaerobic ammonia oxidation, which is eco-friendly removal of nitrogen from wastewater. In other words, green nitrogen removal. So it's a very efficient method of removing nitrogen from wastewater with low energy usage and small carbon footprint without consuming organic carbon. Yeah. Now shall I? Yeah, my screen, is it clear? Like, is it visible? Hello?
No, your screen is not shared.
Oh, yeah, just 1 minute. Yeah, it's not shared. Yeah, now?
No,
Hello.
please share your screen.
Not
ma'am.
visible?
Yeah.
Huh? Now it is visible?
Is it visible?
No, it is not. No.
Yeah, 1 minute.
You're audible, but your slides are not visible, ma'am.
Okay. So it's an efficient method of nitrogen removal from the wastewater, which is nothing but nitrogenous organic matter removal with the low energy usage and small carbon footprint.
Please share your screen.
Yeah, without consuming organic carbon.
Is it visible?
It's coming.
Yeah.
Yes.
Is it visible?
Yeah.
Yeah.
Yeah, seen anlikattaralla.
Yes, visible.
Yeah.
Shall I start?
Yeah. Okay. So anaerobic ammonium oxidation is a biological innovative nitrogen removal. It is becoming increasingly recognized for the application in the wastewater treatment plant. So it was discovered in the wastewater sludge in early 1990s with a unique ability to combine ammonium and nitrite or nitrate to form nitrogen gas. Okay, so this is how the ammonia and nitrite giving back nitrogen. So this process, like how it was discovered and what is its importance, all that I'll let you know. Yeah. So this is a growing demand, and this process is very slow process, and the organism requires not less than 2 to 3 weeks to grow. So we have gone for metagenomic analysis and got the organism identified. Already Muskan has told about metagenomics and how it is done. So here I will tell exactly how we are able to see this organism growing and how it is bringing wastewater treatment or cleaning water rather. Yeah. So this is an aquaponics basically. So the fish waste is rich in ammonia, which is consumed by plants in the form of, which is getting converted to nitrate by nitrosomonas and nitrobacter, and again it is going back into the atmosphere by this NMOX process, which is an anaerobic, as well as aerobic process, which is nitrification and denitrification. So here the main importance here is how this fish is able to survive. The fish is getting the nutrients from the plants. The plants are supporting PGPR. The roots of the plants are supporting PGPR bacteria, which are supplying necessary bacterium for the fish to survive. And the fish gut is supporting all the secondary metabolites or the number of the chemicals which will kill the pathogens and allow only the useful microbes to grow, and in a way helping the fish to multiply, spawn easily, as well as supplying nutrients for the plants to grow. So this is complete symbiotic process. And exactly how this NMOX is working. See, this is an aerobic process where complete oxygen is required, and there is a lot of CO2 emission and nitrous oxide emission. Whereas in NMOX process, there is less carbon utilized, since nitrate is a source of energy, so there is no role of carbon source nor oxygen and the evolution of nitrogen, nitrous oxide or CO2 is totally minimized, or rather we can say the greenhouse gas emissions are totally eliminated. So this is how this is the novelty of the process. And when we have gone for the metagenomic analysis, we have seen that there are a group of bacteria called planktomycetes which are playing major role in this process. So planktomycetes, and yeah, metagenomic analysis proves that there are a set of complete microbes which are cleaning water, but in that we have only the planktomycetes playing major role in this biogeochemical cycle of nitrogen and carbon. And along with the planktomycetes, we have another bacteria, microbia, which is close to planktomycetes. Hence, all three are classified under one class or under one superphylum, PVC, that is planktomycetes, microbial, and Chlamydia. Of course, Chlamydia was the first one to be isolated in this complete phylum. Yeah, so this is the metagenomic analysis showing that by 16S sequence it was identified, the GC count of it, and the complete taxonomy analysis, and the complete bacteria in it, the genus, and yeah. So this is exactly the bacteriological characteristic showing that we have planktomycetes and Akkermansia. This is a member of microbial. So planktomycetes, Akkermania, and Chlamydia are supposed to be included under one class because of their close characteristic feature and having this process called anemones. The next is next thing is what are planktomycetes. So they are a fascinating group of bacteria which are very difficult to, as I said, they require not less than 2 weeks to grow. So, and even after growing them, it was very difficult to tell whether it is a bacteria or whether it is a fungus, whether it's filamentous, whether it is actinomycetes. So only the metagenomic reports showed the clear picture that it's a bacteria, it's having a bacterial characteristic feature as well as the eukaryotic features are having a definite nucleus and a cell organelle called anemosome, having anemox process going on. So that is a distinctive feature of this anemox bacteria, and this is an interesting group for from ecological point of view, from environmental point of view, from bioremediation aspect, and as well it has got a number of bioactive compounds synthesizing, as we are able to see them in a closed aquaculture system, making the plants to grow easily in a cyclic manner. Yeah, this is a setup, and this is the pond. Yeah, this is a setup where we have plants as well as fish growing happily luxuriantly. Yeah, so this is the nitrogen cycle, as I said. So this is anemox, the red one, and the green ones. These are the actual anemox process, the innovative process of nitrification, which is not involving the role of oxygen. It is totally anaerobic conditions. So it has zero carbon footprint and zero greenhouse emissions. The next coming to actually I'll show you exactly how those. Yeah. So there is.
Please conclude, madam. Time is up.
Yeah. There is a historical aspect, like how just because they are difficult to be grown in lab, how it started and who are all the people who have contributed to the studying of these organisms. Have given the list of all the people. So it started from 1973. For the first isolate was done by Staley. Then further it was So it is. In 2001 and 2006, the superphylum was designated calling PVC group of organisms. So at present, this planktomycetes has two classes, the Candida brocchidae and Physisperae. So these are the one which is having anamorph process, the other one which is not having anamorph process. And then I'll just show the video link, like exactly how I have got this identified, or you can see the.
Audio, I don't know.
Hello, is it audible? Hello?
No, presentation is not audible. Your voice is audible.
Oh, presentation. The figures are not clear. Hello. The presentation is not there.
Video is working, ma'am. Video is working.
Okay.
Sharing the kinda tickle button.
Audio from the video is not there, ma'am.
Audio is not there.
In the bottom, 2 check boxes will be there, ma'am. You just click the check boxes in the PPT.
Yeah.
While sharing.
Okay. Was it clear, madam? Hello?
Yes.
Yeah. So that's what. So then, I'll conclude. So that the anamorph is a process for sewage treatment with the least greenhouse emissions. So I thank you for one and all for giving me this opportunity. Yeah.
Madam, what is the status? Like, anyway, as you have shown, it looks like as if, yeah, what is the status of this work? Like,
hello?
What is the status?
Yeah,
like, I can see that it has been restricted to one place.
Yeah.
So if you want to use it in future for bioremediation or something, like, do you have any plans for future?
Yes, madam. That's what I'm trying to apply for BRAC. Last time I've applied for this closed recirculating aquaculture system with nano bubble technology.
Okay, because there are many approaches.
Yeah.
For remediation and all.
Yes, madam.
Do you feel that your technique is better than them?
Yeah, this technique is better, but only thing is the cost wise it is more, but the oxygen, what to say, the requirement for oxygen is less, but cost may be more, and the plus
cost effective.
Yeah,
Like,
it is cost it is cost effective.
People want to go for utilizing your technique.
Yeah,
it should be cost effective, especially in countries like ours.
Yes, it is. Yes,
maybe you can see the aspects where you can reduce the cost of it. You
Yeah,
can
that's
compare with other things.
That's what using this nanobubble technology probably it should come down. In countries like Netherlands, it was Netherlands, Australia, they are
because they can manage, but our country it is a little tougher, I believe.
Yes. So that's the cost is main. Otherwise, it is highly eco-friendly.
Okay.
Yeah. Okay. Thank you.
Revati, next.
First of all,
Thank
very
you,
good evening to all the dignitaries. I am D. Revati, research scholar under the supervision of Professor B.S. Anuradha at Chaitanya Deemed University, Microbiology Department. Is my screen visible, sir?
Yeah, it is visible. You can continue.
So today I'm going to present the topic study on antibiofilm activity of Tinospora cordifolia plant leaves and Pseudomonas species. First of all, biofilms are the communities of microorganisms which are attached to the surfaces, and they play a significant role in the persistence of bacterial infections. And the bacteria which is present in the biofilm show high degree of resistance antibiotics compared with the planktonic bacteria, and this makes it difficult to treat the infections related to them. And plants are the best sources of antimicrobial agents with relative or no side effects when compared with the chemically synthesized one. And this is the reason nowadays antimicrobial agents from the plant sources are commonly used. And this is the slide which explains about the steps which are involved in the biofilm formation. First, adhesion of the planktonic cells to the surfaces, and next, formation of monolayer, which results in the formation of slime, and it develops into microcolony. The next, multi-layered cells are formed. The next, mature biofilm microcolony develops into a mature biofilm. Then next, detachment of the cells, then they are converted back to planktonic cells. This is the cycle which explains about the biofilm formation. And the present work is an attempt to screen for antibiofilm activity of Tinospora cordifolia plant leaves and Pseudomonas species. And phytochemical screening have been performed after extraction process. Then coming to the Tinospora cordifolia, this is the plant which is grouped in family Menospermaceae. Common name Boducci. Useful parts are leaves and roots. And therapeutic uses include anti-diabetic, antiperiodic, antimicrobial, antibiofilm, and immunomodulator. Recent studies have shown that Tinospora cordifolia has antiviral activity, which is used in COVID-19 treatment. Materials and methods. I have collected the leaves of Tinospora cordifolia plants, and they are shade dried and stored in the room temperature. Then the leaves were made into coarse powder, and I have started the plant extraction process. Maceration method was employed to make an extract, and methanol is the solvent which is used here. And the excess solvent from the filtrate is evaporated, rotary evaporator, and the weight of the compound which is obtained is calculated, and that is dissolved in dimethyl sulfoxide for future studies. And the bacterial culture I have used is Pseudomonas species, and it is gram-negative bacterium, which is known to cause infections in blood, lungs, and other body parts, and even after surgery. And before screening for antibiofilm activity, I have performed biofilm production detection process, in which inoculum preparation, fixation, staining, and elution are the major steps. Then next, I have performed phytochemical screening for the obtained extract. It was found that some of the alkalides, diterpenoids, and some other steroids have been identified upon phytochemical screening. And before performing antibiofilm activity, I have first performed antimicrobial activity to check whether this compound is showing antimicrobial activity or not. So fresh culture is inoculated into the Muller internal plates, and the extract to be, the compound which is to be tested is loaded into the wells by punching the agar plates using chlorine. Then I have added extracts into the wells, and these plates were incubated at overnight at 37 degrees centigrade. And the clear zone, which is formed after incubation, surrounding the antimicrobial activity. Then next, I have performed minimum inhibitory concentration. So this is performed by microdilution methods, and I have added extracted different concentrations ranging from 0.1 mg to 1 mg. Then minimum inhibitory concentration was also being calculated. Then next I have performed the assessment of minimum biofilm inhibitory concentration. This process also have performed for detection of biofilms inoculum preparation, fixation, and staining evolution. Then later I was found that results and discussion. The method involved the administration of methanol extracts of plant leaves obtained by maceration procedure, and the zone of inhibition after antimicrobial activity was calculated and was found to be 23 mm in diameter, and the minimum inhibitory concentration values of Tinospora cardifolia plant leaves on Pseudomonas found to be 300 micrograms, and the minimum biofilm inhibitory concentration for Tinospora cardifolia or Pseudomonas species was found to be 500 micrograms. And conclusion, studies revealed that plant leaves exhibit antimicrobial and antibiofilm activity, and upon phytochemical screening, the presence of bioactive compounds like triterpenes, formalins, furanins, tannins, saponins, and steroids have been reported upon phytochemical screening, and further GC analysis will help to know to identify the compound extract and also to know the molecular weight of the bioactive compounds. I thank Professor C.H.B. Purushottam Medicine, founder and Chancellor, Chaitanya University. I also thankful to Professor L.B. Lakshmikant Raju, Vice Chancellor, Palmer University, Mahabubnagar, for their constant support and facility. That's all.
Revati. Yeah. Revati, actually, like why you have selected only methanol? What is your criteria for selecting methanol for extraction? You can use some other solvent.
Yes, madam. I have checked with other solvents also, but the polarity of the compound is similar to this one.
See, you can't go only for one, because it's not that there is only one type of phytochemical. No, there are groups, as you have already shown in your thing. There are alkaloids, there are lignans, there are different type of compounds which are present, terpenoids and all. And one more thing, so you should try different solvents and you should see that where you are getting the best results. Other than that, like what are exactly, there might be reports, earlier reports, no? Because see, these are alkaloids, for example, if you say alkaloid is a group of compounds. So like, because each plant has its metabolic fingerprint. So they have different kind of compounds which are present in them. So you can't just go for grouping that way that these are present, these groups are present. You have to be very specific about what compound is present in the plant. And what is the mode of action for this? Okay, go that way you have to go. Fine. Okay. So it will help you to go further for your studies.
Yes, madam.
Yeah.
I want to go for GC analysis.
Yeah. No, that before that also you have to see.
Even I would like to ask the same question. You have said that you have got many active compounds like tannins and terpenoids, but you have not shown any biochemical reactions through which you have identified these chemical compounds. That would have helped in the materials and methods section. That's what madam is asking. So there are some procedures to understand what are the chemical compounds you have identified as the active principles. So that part you should have projected here if you have done. Because you have done only, you are presenting that these are there. So that part representation is missing. That's what I guess.
And above to that, like see, you just go for the previous literature because you have to be very specific about the compounds, Then you can go for HPLC analysis and all to find out exactly if that is present in your extract or no. Because only biochemical acids not might be
one more thing is like you have to go for GC-MS analysis, not just GC analysis. That is
and LC also, side by side LC also, because GC goes only for volatile compounds. So yes. LC also you have to go for because you can detect the compounds which are not non-volatile compounds also.
And so, you want to know the molecular weight, you have to go for
that's what, there are so many things you have to take.
I have done screening process and phytochemical screening process. And further work I have to continue.
Yeah, anyway, because maybe you have still to work on those aspects.
Thank you.
Thank you, ma'am. Next, I invite Shiva Kumar Singh, sir, to please present your presentation.
Good evening, everyone. Dignitaries, Department of Botany, Department of Microbiology, myself, Dr. Shanthi, Faculty of Botany. Sir, my slide is visible to you all, sir?
Please go for full screen. It's better.
Slideshow. Slideshow.
But your voice is not clear, Dr. Dr. Singh. Your voice is not very clear.
Is it visible now?
No,
Sir, a slideshow చేయండి. Slideshow. Make it slideshow.
keep your microphone also near you so that the voice is louder.
Sir, there is some problem, sir. Can you present afterwards? Meanwhile, you know.
Audio is not clear, sir.
Sir, audio is not clear. Can the next person, you please wait for the next this thing. Let someone else present, sir. You rectify your problem.
Sir, is my slide is visible, sir, right now? Already I
No,
shared.
sir. Slideshow రావట్లేదు, sir.
I shared, sir, already.
No, it's not coming. Reshare it, sir.
Your slides are not moving and it's not gone into
Your
slideshow.
audio is also not clear, sir.
So meanwhile, we have another participant waiting.
Yeah, that is better.
We'll move with the other participant. Veena, if you're ready, can you please share your presentation, please? Anjanelu sir, please unmute Veena and allow her to share her presentation.
I've done it. I made it.
Yes, madam. Yes, good.
Okay, sir.
Yes. Is it visible, ma'am? Hello? Hello?
Yes,
It is
yes,
visible.
yes.
Yes. Good afternoon to one and all. Thank you for giving this opportunity, sir. So this is Veena, a research scholar in microbiology, Chaitanya D. Muthubi University. My supervisor, Professor B.S. Anuradha, ma'am. Is it clear, sir? Please.
Yes, ma'am.
Yeah, thank you, ma'am. My topic is on about characterization of amylolytic yeast from the various natural sources. That amylolysis process is a conversion of starch into sugar by the action of amylase enzyme. Yeast converts the sugar to carbon dioxide, alcohol, energy. Then the starch or amylum is a polymeric carbohydrate found chiefly in seeds, fruits, tubers, roots, and stem of plants, notably in corn, potatoes, wheat, and rice. Amylum in important foodstuff is and used. The amylolysis hydrolyze the starch molecule into glucose and maltose. Glucose, maltose, and dextrin. They have many applications in bread and baking industry, starch, liquefaction, and the saccharification, textile, paper industries, detergent industries, analysis in the medical and clinical chemistry, sorry, food and pharmaceutical industries. Starch amylolytic yeast strains isolated from the starchy soil, flour mills, environment, flour market, cassava farms after harvesting the transformation of tubers. Next, I used different types of the materials and the methods in this for the isolation of amylolytic yeast. The YPD or YEPD medium, that is yeast extract peptone dextrose media, is used to isolation of amylolytic yeast. In that medium, streptomycin is added to it is a yeast. So some bacteria for the inhibition of the growth of the bacteria, I used this streptomycin. The medium was stabilized in the autoclave and streak. I used streak spread pour plate techniques were employed to obtain the well separated colonies. Amylolysis amylase clearing zone assay medium is used. In this medium, soluble starch 0.5 grams and agar-agar 15 grams. This content were dissolved in the 1 liter of acetate buffer. I maintain the pH is 5. How this I isolated these amylolytic yeast from the different species. For the isolation of yeast were collected some of fruits. I took many types of the fruits. Example, that is a banana, apple, custard apple, and the soil sample, leaves. But so many fruits and leaves materials I took, but I got these amylolytic yeast from the few of these substances. That is the flowers, garments, toddy samples, and brought to the laboratory. In case of soil samples, some different types of the soil sample I took. The suitable serial dilution were made. These plates were incubated at 30 degrees centigrade for 24 to 48 hours. The ripened fruits were washed with the water, remove and adhere soil and dust particle. They were cut into the small pieces, placed in the aseptical and YEPD medium plates were Exora species are collected, washed with the 70% of alcohol to remove the contaminants, to remove the bacteria and some of other fungal cells, and placed in the aseptical in the YEPD medium. There is a yeast extract peptone dextrose medium. These are I isolated by amylolytic yeast from the hibiscus plants and the toddy samples. These are I isolated amylolytic yeast from the different natural sources. Then screening of amylolytic yeast. All the isolated yeast strains were examined for their amylolytic activity as follows. All the isolated yeast strains were grown in the flask, flask containing 50 ml of sterile amylase production medium, and were inoculated with the 1% of yeast cell suspension. Then the flask were incubated for 48 hours at 25 degrees centigrade. At the end of the incubation period, the content of each flask were centrifuged at 8,000 RPM for the 15 minutes at 4 degrees centigrade and filtered through the Whatman filter #1 filter paper. The superintendent collected the crude enzyme extracted, which is used to determine the enzyme productivity of the strain through the measurement of enzyme activity. For the measurement of enzyme activity, then 20 ml of aliquot were sterilized. Medium were poured aseptically in each sterilized petri dish. The medium were allowed to cool. Solidify 1 cup was made per each plate by the sterilized rock borer. 0.1 ml of crude enzyme extract was introduced into the cup, and then the plates were incubated for 48 hours at 30 degrees centigrade. At the end of incubation period, plates were flooded with the iodine. For the potassium, flooded 0.5 grams, and the potassium iodide. This is used for the production of amylase enzyme is indicate the by the existence. This is a screening of amylolytic yeast that is isolated by the observed by the zone of inhibition around the colony. Then result and discussion. During the growth of on agar plates containing starch as a substrate, amylolytic yeast formed a large digesting halo around its colonies. This is its colonies after straining with the iodine vapor, showing that this yeast secreted starch digesting enzymes. The time course of amylase secretion and starch degradation by the yeast during the batch culture in the medium containing soluble starch. This is result. Next, my conclusion is yeast secretes one glycoprotein with the amylase activity. When grown in the presence of starch, amylase was purified and exhibited high affinity of starch. However, further study will be required to Yes, sir, it's over. Determine the structure of this amylase and the role of the carbohydrate moiety in its conformation stability. This enzyme may be good system for the studying of role of carbohydrate, for the studying of carbohydrate during the synthesis and secretion of the glycoprotein by the yeast as a possible model to understand the relationship between the glycosylation and enzymatic activity in the yeast amylase. Thank you, sir.
Thank you, Veena. All the very best.
Actually,
Dr.
there are many reports. Can I ask one question? There are
Yes,
many
ma'am.
reports of amylase production using like fungal cultures and all. So what is the novelty of your work?
So, ma'am, this is industrially important amylolytic yeast, ma'am. This.
Are there no reports of yeast use for production of these amylase production or something which is of industrial significance? There are reports, I think.
Ma'am, I didn't get, ma'am.
No, there are reports of yeast producing this amylase, no?
Yes, ma'am.
Earlier reports are there. Then what is the novelty of your work?
What is now? మా ఫ్రెండ్కి డిక్లో పెట్టి అంటే ఒక దగ్గర
Yes, like because see, if the already it is a reported work, there should be some novelty, isn't it?
Yes, ma'am. Yes.
Also, Veena, just with the zone of hydrolysis, how do you want to progress? Because so many fungi will show this amylolytic activity. You know that is just qualitative analysis. You have to first find out if this is having enough amylase activity, which can be actually studied further, right? How many cultures have you screened?
Means I took many types of samples, ma'am, but from few of the samples I got amylolytic yeast, ma'am, just from ripened fruits.
Yeah, but you have not quantitated, right? Well, you have not studied if there is enough amylase activity that you should study, because naturally there are so many microbial strains which will produce, right? What is the specialty that, like Madam has also pointed out? Is there anything different you want to do? You have to think.
Ma'am, that it is absorbs the starch which is degraded into the alcohol and which is degraded into the alcohol and carbohydrate, that is the glucose, dextrin, maltose. So this starch is the cheapest material by the absorption.
Yeah, I mean, in the conclusions you have said that the enzyme is purified. Have you purified the enzyme?
Yes, sir.
What are the methods you have used for purifying the enzyme?
Sir, your voice is not clear, sir. Please.
How have you purified?
How did you purify the enzyme?
Just I got only pure that by using of just by the sprinkling of iodine.
That is not. You have to go for purification strategies.
Then otherwise, how will you know the assay? How did you do? I mean, just assay without purified enzyme. It's partially purified enzyme. It is not right.
Okay, sir. Okay.
And did you identify which one it is, which the organism, the yeast, which one it is?
Sir, this is yeast cells, ma'am. But
yeast, it is a group, you know. You have to identify exactly which one it is.
It is as
see, you have, there are some loopholes. You have to see first, like how to take care of all those things.
Yeah, and also she told it is in a glycoprotein. What are the methods through which you identified it is a glycoprotein enzyme? That also is a very important proof which you need to provide.
Just I identified it is a amylolytic yeast, ma'am, but I not concluded, means that is the next of my works, ma'am. Yeah, I did not complete, means not confirm, ma'am, that is a special
yeah, but
organism.
Veena, this is a MSc practical class actually. What this is simply finding from an MSc class, from a practical class, you know. So maybe you have to think how what more you can do in this area, because just by showing amylolytic the zone of inhibition, it's not sufficient.
Ma'am, this amylolytic enzyme which is present in yeast, this is present in where my next work is it is present in where either maybe plasmid or which ma'am.
You mean it's not
a
a
generic term, you know.
I mean, it's not a general activity of the yeast.
That's what.
You have that's a lot of things. You have to read a lot of things about this. Yeah.
I think she requires a proper planning first.
Yeah. Yeah, you have to talk to
Otherwise,
your guide or whoever who is guiding you. You have to sit with them and understand what exactly you are doing with something which is already known, you know.
Okay, ma'am. Okay, ma'am.
Dr. Shivakumar sir.
Hello, sir.
Sir, please present.
Am I audible?
Yes, sir.
Now slide is visible, sir. Sir.
Slideshow button, sir.
Yeah, now okay.
Sir, by selecting single slide, you can continue, sir. No issue.
Okay. So good evening, everyone. This is one of the fundamental work on silver nanoparticle synthesis using plant extract. As we know, the biosynthesis of nanoparticles using plant-based extracts from non-polar to polar, like hexane, petroleum ether, chloroform, ethyl acetate, methanol, and water, and so on. So here, basically, we used to practice this type of activity. But in case of Vitex nigundo, this plant is one of the very rare plant. And moreover that on this plant, we have got one different variety where we can found like a brown type of variety. It is available in particular Telangana and Karnataka borders. There the leaves are showing a different types of chlorophyll content. Particularly there we have concentrated on this silver nanoparticle synthesis. So that here particularly we have concentrated on aqueous extract as we fundamentally practiced it. So here this Vitex nigundo showing a rapid biosynthesis of silver nanoparticles within 10 minutes. And that too, the percentage of nanoparticles also we have calculated near to 11, 1 to 11 and 11 to 40 and so on. Their percentage also we have calculated, and as well as we also characterized the nanoparticles at one side and another side. And we also concentrated on solvent-based synthesis. Here we only presenting that the aqueous-based nanoparticle synthesis, where this on the basis of the solvent-based synthesis, mainly we are concentrating on exactly what type of hydrochemical is going to be responsible for synthesis of the silver nanoparticles. Also, we are concentrating. So here, just we optimizing the concentration levels of the nanoparticles, so that this plant containing a huge amount of tannins in their bark and as well as their root system. But we just compared the bark to the seeds in phytochemical studies. But in case of this silver nanoparticle synthesis, we have selected the seeds. In seeds, so the percentage of biosynthesis for nanoparticles and compared with the bark also we have studied. But here we only presenting the seeds and their optimization of the concentration levels and their extracts we presented here. So the common method we followed, like this is a Vitex nigundo brown variety. This is about methodology. As methodology, here we have two types of methodologies. We have followed. One is a fresh leaf method. In fresh leaf method, in biosynthesis of nanoparticles, we have found some of drawbacks like chlorophyll content, even water content, other mucilaginous contents also responsible for the synthesis of these types of nanoparticles. So that is why in case of the shade dried and even after, as we following the phytochemical methods, in those methods we have got a good optimization, and the characterizations also we have found very clearly, so that in case of here, like this method, schematic diagrammatic method, in using the fresh leaves or seeds, whatever the plant material we are targeting there, like just taking a weighing of a particular leaves according to their optimization levels. Then afterwards we have to take the filter, that is about aqueous filter, and there the silver nitrate should be used in case of 1 millimolar for the 100 ml of water, and that should deionized water. And there 10 ml of Vitex nigundo extract was added into 90 ml of water aqueous solution of 1 millimolar silver nitrates. Then incubation should be required. In case of this plant, only at normal room temperature, and even there with rapid color change we have found. Of course, the color change may not be the synthesis of their particular nanoparticles, but that should be proven by using the UV, FTIR, and as well as XRD and TEM. And this picture shows the experimental changes, and it's about the silver nitrate solution and plant extract, and this is about the synthesized nanoparticles conical flask. And these are the characterization of this particular graphical structure of the UV, XRD, and this is about the TEM. In case of TEM analysis, clearly we have classified the nanoparticle type sizes about from 11 to 41 nanometers. And the shapes also, different shapes also, we have characterized about the 25% of the particles 11.09 nanometers, and 35% of nanometers related to these 30 nanometers. So these are about the classifications of the nanoparticle sizes. In case of the anti-dermatophytic activity, why we only concentrating on these nanoparticles using with anti-dermatophytic activity means we here also we compared the phytochemical, whatever the crude drug we have extracted with the saxolate apparatus with the different solvents, like one ethyl acetate extract we have selected, and at the same time ethyl acetate related to the silver nanoparticles also we have selected. In this direction we have compared the anti-dermatophytic activity of the particular nanoparticles we have studied. And here ultimately what we conclude that here within 10 minutes with the normal room temperature, the silver nanoparticles are synthesized. Moreover, that from this plant and this variety, the nanoparticle synthesis with the antidermatophytic activity is a first report from this area. Thank you.
So, Dr. Singh, thank you for the presentation.
Yeah, here I'm getting some difficult.
Yeah, in the graph you showed in the end, right? What can you show the graph again, please? The graph where you were showing the zone of inhibition, no?
Yes, ma'am.
I think you're unable to share. It's fine. So
Yes,
in the zone, the silver nanoparticles showed the least, looks like they showed the least zone of inhibition, right?
Yes, yes, madam.
Then how do you want to use these nanoparticles? How do you want to say that these are competitive enough? Because in the market already we have Silver X, and so many silver-based ointments. So how do you think this study will be useful?
Of course, there what I shown in that graph, silver nitrate is showing as a least activity. Silver nitrate is selected as a negative control.
Control. Okay.
Yeah,
Which
that
one
is
is
showing,
yours?
that is silver nitrate. Whereas my silver nanoparticles using with aqueous extract showing triplet of the activity. Madam.
Triple, huh? Okay.
Yeah.
In that graph, the label was not clear. That's why I wanted to
Yeah,
know.
actually, madam, this last side, whatever you have seen, that shows the negative control. Negative control you used silver nitrate, and another one fluconazole also I used. Antidermatophytic compound is there now already in standard compound. Fluconazole also I have compared, but this silver nitrate is not crossed with fluconazole. 50 or more than 50%, it has crossed.
Okay.
Yes.
Did you do any stability studies with your nanoparticles, like how stable they are?
Yes, madam. Stability studies means one of our material science professor is helping in that department.
Because
We
that
are
has
not.
to be done.
I am only expert with the phytochemical analysis.
Yeah,
So
because that is also a part of that, no? Your study, like stability studies are must for that.
Yes.
And one more thing, I have a little bit this thing with your presentation, because you shouldn't put so much of material in one slide. Like each slide is going, it looks very clumsy,
Yes, madam. Of
so
course.
that is really a part of the presentation that you have to take care of it.
Okay, definitely. In case of stability, madam, what we can do in laboratory when after getting a color change is physically we observe, and secondly we have to be keep such a material or petri plates or conical flasks or test tubes, whatever we use now, we have to keep for the long time, like 15 days, 20 days.
No, there are certain protocol,
Yeah.
how you have procedural details are there, how you have to go for stability study.
Yes, madam. Of course, in that
Like that.
aspect we should have to follow.
Okay.
Okay, thank you very much, madam. Whatever fundamentals available with us.
We thank
Now
all
we
the
are going to-
delegates who have participated in the paper presentations. We had a very good session. I thank all the jury members also. Next, we have the poster presentations.
So now the posters are scrolled on the screen just to be presented there. Based on that slide, judges will be gives the judgment. If anyone is enthusiastic to present their presentation regarding that poster, raise your hands in the chat box. We will give you 2 minutes time. Hello. Yes, Chandana. Can we go the another slide, Chandana? Second poster absent. Mahesh, are you there?
Unmute చేయలేదు కదా.
చేయండి, unmute చేయండి. Chandana, unmute yourself and talk.
Hello everyone. I am Chandana from Telangana, Hyderabad. I am a student of Raja Bahadur Venkatarama Reddy Women's College. So actually, I'm not a student from science background. I'm a commerce student, but enthusiastically, like I love to just I'm very much interested in science. So just I participated here and took taken up the topic here, cellular I mean, cellular microbiology and system microbiology. I have written a small mistake here. Instead of microbiology, I have written microbiology. Please forgive me for that. So, coming to cells, I want to just give a small introduction regarding the cells and the definitions of the topics which I have taken. The cells are, as we know, cells are the basic building blocks of every living organism in the world, or even in the earth also, because without a cell there is no such a thing exists. So we can say that cell is a very important element of the living beings which exist on the earth, and not even a living being, but the non-living beings are also made up of cells, but they are called in another way. So when we are coming to the living beings, it is again we can say that there is a cellular microbiology. So I want to just give a small definition of cellular microbiology. Actually, it means the branch of science that deals with microorganisms, and hence.
Please conclude, Chandana. Please conclude.
Oh, okay, ma'am. Okay, ma'am. Just to give me 1 minute. I can just, ma'am, can you share my e-poster? This is not my e-poster, ma'am.
Okay,
Your
next.
time is over.
Okay, thank you for giving for the opportunity.
Mahesh,
Yes, sir. Yes, sir. Can I share my screen, sir?
I already
Yes,
shared.
sir.
You just explain.
No. This is not my screen, sir. This is somewhat to another person, I think. This is myself. This is myself. So myself, Mahesh. So good evening to all delegates and the coordinator and the organizers of the Palamur University. So my poster is design, development, and characterization of fluostatin. Sir. It's gone, sir. Hello, sir. My poster is gone. This one only, sir.
You carry on.
Fluostatin pulse. Yes, sir. Fluostatin pulsatile capsules. So this is mainly we are formulated. Why? Because this dosing is required at the night time only. The drug will be released from this dosage form. Why? Because in the night time the cholesterol synthesis will be high in our body according to chronopharmacological behavior. Why? Because this whatever the food particles is converted, carbohydrates are converted into fat at the night time, and the enzyme activity will also high. So that's why this pulsy capsule is best helpful for the reduction of the conversion of or synthesis of cholesterol in the peak time in the chronopharmacological behavior. That's why
this is best suitable formulation for the reduction of the conversion or synthesis of the cholesterol in the night time, and where the dose is required in the night time only for the patients who are suffering with higher cholesterol levels. And here we are using the fluvastatin, which is having the short half-life. They are best helpful for the formulation of this pulsy cap. Here I am using some hydrogel plug. After 5 hours of taking the medicine, the hydrogel plug will be swells by absorbing the moisture. Yes, okay, thank you, sir. And that the drug will be released and it will shows its pharmacological action there. They will reduces the cholesterol synthesis, that is conversion of acetyl-CoA and cholesterol, where the HMG-CoA reductase will be stops the conversion of malonic acid, sir. This is my post presentation. Thank you, sir. Thank you, sir. Thank you very much.
Shravani Sharma is there. Raja Reddy is there. If you are there, raise your hand.
Very good evening, sir. Sir, am I audible, sir?
Yes, audible.
Okay. A very good evening to all the dignitaries. First of all, I would like to introduce myself. My name is Shravani Sharma and I'm studying MSc first year from the Palamuru University. So today I'm here with the topic with strategies of antiviral therapy on viral infection diseases. So antiviral therapy. Antiviral therapy is one of the most emerging therapy, and it is a most emerging. So please can you give me the 1 minute, sir?
Yeah, I will give.
Yeah, sure. Thank you, sir. So the antiviral therapy is one of the most emerging concept in virology, and antiviral therapy is also under the principle of gene silencing, and it specifically target the viral DNA on RNA, and it helps in treating the viral infectious diseases. And the antiviral therapy is
Next slide. Next.
So many things ఒకటి దాంట్లో పెట్టిన ఫాంట్స్ చిన్న ఫాంట్స్ అది.
Good evening, all the jury members. I'm Mercy, who is working as a degree lecturer at TSW RDCW Medak. I'm pursuing my PhD under the guidance of Dr.Shanthi Ajmera. So this is the quantitative work which was conducted. The qualitative work is yet to be conducted. So this is the biosynthesis of silver nanoparticles, and we have also characterized their antimicrobial activity from the thermophilic fungi. There are 4 thermophilic fungi which are taken: Humicola gricia, Mucor mihi, Malbranchia sinomia, Cytalidium thermophilium. So out of these three, two of the thermophilic fungi have shown the synthesis of silver nanoparticles when silver nitrate is added into the media, and their characterization is also been done by 18S rRNA. They also showed antibacterial when disk diffusion method was performed, and they showed greater antibacterial property using this silver nanoparticle extract than the crude culture filtrate. Around 30 mm inhibition zone was seen to inhibit Staphylococcus aureus, and to inhibit they inhibited Proteus and showed 25 mm zone of inhibition, and in case of E. coli, the Cyatilidium showed 13 mm zone of inhibition, and further characterization is still under process. Thank you.
Okay, good. Next. Shravani Sharma helping.
Good evening, everyone. Am I audible, ma'am?
Yes, yes.
Yes, ma'am. Good evening, everyone. I'm Pujita, a student of Bhavan Vivekananda College. My poster is entitled as A Review on Globally Emerging and Re-emerging of Animal Viruses. Over the years, a number of novel pathogens continue to emerge in humans and animals. In Southeast Asia, a new human viruses which led to devastating epidemics like SARS coronavirus and highly pathogenic avian influenza H5N1 virus was discovered and reported since 2007. Chikungunya is another type of virus which is related to group A arboviruses, and it occurred between 2005 to 2006 and in French Reunion Island in the Indian Ocean, followed by several outbreaks in other parts of the world, such as India and Southern Europe. Coronaviruses in pigs is another example, where porcine epidemic diarrhea virus, porcine delta coronavirus, and swine acute diarrhea syndrome are emerging and re-emerging coronaviruses. It was first recognized in China in 2010 and rapidly spread worldwide, including at least 10 states in US in 2013. Emerging and re-emerging viruses of honeybee, Apis mellifera. It was first detected in US in 1987 and quickly spread to Mexico and Canada. The worldwide introduction of ectoparasite mite varroa destructor, beekeepers encountered difficulties in maintaining their colonies. They usually live for 2 months but can hibernate between the sclerites of adult honeybees. So, the various approaches for combating these viral diseases are selection of tolerant bee species, RNA interference, and prevention of entry of new pathogen. Thank you.
Okay. Next slide. Vijay Kumar, are you there? Vijay Kumar.
Naman, you want to present?
Yes, sir. Good evening. My name is Naman, and
I have made a poster on the clinical infection and vaccines. As we know, clinical infections are a major infections caused by microorganisms. They are a major cause of death and disability around the world, and they affect the public health and economy of major developed and developing countries. That's why it is a major concern for the world leaders and health policy makers. To control and contain these outbreaks, viral outbreaks, vaccines are proven very effective in controlling death, disease, and disability, and they are even cost efficient. We can take the example of the outbreak of polio virus, which was contained in the by the polio vaccine, and as the new variants and viruses are emerging, so is our medical base to uh, yes, and we are using functional genomics, genetic epidemiology to even further help us research and studies. Thank you.
Sagar. Sagar, are you there?
Yes, ma'am, I'm there. I'm there. Good afternoon, ma'am.
Start.
Start, Sagar.
Yes, ma'am. That time I got muted. So my topic is good afternoon, judges. Sorry for that. My topic is study on the action of antiviral drugs. As we all know, in virology, antiviral therapy is the most exciting place because of which they reduce the amount of virus load in our body and risk the transmission of the viral infections and protects the immunity system. There are many very antiviral drugs which have been approved by FAO like ribavirin, acyclovir, valacyclovir. But because before making, there should be some strategies based on it, like how they should be prepared. So there can be two approaches. Either they can be targeted, like one approach can be of targeting the virus function, which includes inhibiting nucleic acid polymerase like DNA dependent DNA polymerase, RNA dependent RNA polymerase, DNA dependent RNA polymerase, and inhibiting proteases, integrases, and neuraminidase at enzymatic level. And the other approach can be like targeting the cellular function that depends on the virus particle of the which the virus particle is dependent on. And in this table, as you can see, there are some antiviral drug which I named before and their functions. Ribavirin, which is used to treat RSV function, RSV infection, hepatitis C, and some viral hemorrhagic fevers. Acyclovir, which is used for the treatment of herpes simplex herpes simplex virus infection, chickenpox, and syphilis. And valacyclovir, which is used to treat the outbreak of herpes simplex, or we can say herpes zoster. But what is the mode of acting these antiviral drugs? In the action period of antiviral drugs, there are many things happening like enhancement of resistance towards the infecting virus. They suppress the inhibition of. Yes, ma'am.
Conclude.
Okay, ma'am. Then one example we can see of remdesivir, which is a nucleotide analog, which when administered get phosphorylated into triphosphorate form of remdesivir. That is the mechanism which we can see above. Yeah, thank you, ma'am. That's
Likit Bharadwaj, are you there? Likit.
Yes, ma'am, I'm here.
Yes, you continue.
Yes, good evening, everyone. My name is Likit Bharadwaj, and I made a poster on the comparative study of the transmission of zoonotic viruses and that too, in particular, H5N1 influenza, MERS COVID, Ebola virus, and Zika virus. As we know, zoonotic viruses are viruses that are transmitted through animals. And in our case, H5N1 influenza is spread through birds. MERS is spread to camels. Ebola virus is spread through bats, and Zika virus is spread through mosquitoes. So I have done a study where I took the peak times of these diseases and I compared the number of cases to study what the carriers, I mean, how the carriers will be affecting their transmission. What we found out is that Ebola virus was so contagious and dangerous because it was able to achieve so many different kinds of carriers, not just going through bats, but also through primates. And what else we found is that yes, sir. And what else we found is that because of zoonotic viruses, we have a lot of contagion going around, and it might be beneficial for humanity if we understand how these transmissions are going to be going, and that way we'll be able to deal with future cases.
Thank you all the participants and presentees of today's oral and poster presentations are completed.
Yes.
Please on video, everyone on videos. We have to take screenshot.
All participants are requested to unmute their videos, please.
Congratulations, Shanti ma'am. Indeed, it is a successful program. All of them have presented very well.
Oh, no, there is something wrong with my video. I don't know. It's not coming. There is some issue, I think.
No issue, ma'am.
Yeah, I don't know. My way, like from my side, maybe the laptop has something, or I don't know. Like I'm unable to do that.
Actually, okay, ma'am, we will add your photo.
I don't know. Actually, I tried from the beginning. There is something wrong. I don't know why it is not taking.
It's okay, ma'am. I'm very thankful to you all panel members giving your valuable time for us to judge the all presentations. Vote of thanks by Mercy. Are you ready? Unmute yourself, Amma. Unmute.
Yes, ma'am. I thank all the jury members for taking your time and joining us, and for giving us your time and valuable suggestions to all the presentees and correcting them so that they can move forward with their research. Thank you so much for giving out your time for us, ma'am, all the jury members. And we also thank all the delegates for your active participation and for taking your time. We look forward and we want you to continue your research well. And I also thank Dr. Shanthi ma'am for conducting this webinar successfully. Thank you all the technical team also for the support you have given us. Thank you so much, everyone.
We will meet again tomorrow morning, the another session. It's not ended, okay? Everyone please participate tomorrow also and give your feedback. That is the energy to us. Okay, thank you, one and all. Thank you.
Okay.
Madam and team.
Ma'am.
Thank you.
Okay, congratulations, Shanti Priya. It is
Thank
really,
you, ma'am.
yeah, it's nice.
It goes to all team of our
But
department.
anyway, you have taken the initiative, no? So it's you deserve that.
Thank you, ma'am. Next time you are the speaker, ma'am. Archanagiri, ma'am.
Yeah, because actually my area is a bit different from yours.
Yes,
So
that's
because
why
of
we
that,
are not called you, ma'am.
yeah,
Yes,
I
we
know.
pick up the Sharmista, ma'am.
That's why it's
Yeah,
nice.
yes,
Okay, anyway. Okay then.